Okay. Good afternoon, everyone. And let me first thank Professor Alok Bharti for inviting me to this talk in the Conference of Pharmaceuticals and Nutraceuticals for Cancer and Other Chronic Diseases. So today, we'll be talking about it's fine. It's okay now. So I'll be talking on recent advances in brain targeting using polymerasomes or bacocytes and lauric acid and nanotechnology update. With me is Dr. Kritika Goyal. She's sitting here. She's also one of the co-authors of this talk. So, first a brief about the nanocarriers for drug delivery applications. You have a pointer here? Okay, fine. Okay. So, okay, no, there's no problem. Okay. So, you must be all knowing about liposomes. Liposomes are basically lipid bilayers which are used for drug delivery applications, and many of the FDA-approved drugs are being included in liposomes for cancer particularly. Then we have polymerasomes. Polymerasomes are polymer-based structures, whereas liposomes are lipid-based structures, right? The difference between them two is, the main difference is the polymerasomes are more stable, robust in comparison to liposomes. And you all must be knowing about the polymer micelles, where we have got the hydrophobic core and then we have hydrophilic structure, and we can embed both the types of hydrophilic as well as hydrophobic drugs. And another one is the dendron, where you have got leaf-like dendrons attached to the center core, and these structures which are here, they can in fact take lot many drugs attached to these at these places. So the drug loading capacity is also enhanced in case of dendron. In polymerasomes, the drug loading capacity gets very much enhanced because these are the polymeric vesicles. So why we get different type of shapes? It's all because of self-assembly of amphiphilic polymers. We get nanocells if the ratio of hydrophobic and hydrophilic. We have got largely hydrophilic portion and less of hydrophobic portion, whereas in worm-like nanocells, we get equal amount of hydrophobic and hydrophilic structures, whereas in polymerasomes, we have more of hydrophobic and less of hydrophilic structures. So, what are the scientists looking for nanocarriers, which is very important. Nanocarriers have to be safe because when we talk about nanocarriers, these nanostructures can channel become antigens in the membrane, where they can create many toxic effects. So the safety is a great concern. Then we talk about biocompatibility. You know, now I'm much older. I've seen many students, many of you are still here. Whenever they say the slides about cytocompatibility. Cytocompatibility is not biocompatibility. So biocompatibility, there is a procedure ISO 10993, where we talk about nanostructures or anything like with not nano also, we have to follow ISO 10993. Then in that, there's a huge list of biocompatibility of any material who goes into the body. Then the last is easily eliminated. The nanostructures are to be eliminated. For example, in the 90s, there were a lot of work going on gold nanoparticles, and people used to publish thousands of papers on gold nanoparticles, but later on, the nanoparticle size of gold increases 5 nanometers, it cannot be flushed by the kidneys. So, and then the structure, whether it's circular, whether it is any star shape, that also matters a lot. So, we have to safety is a big concern as long as the nanostructures are concerned. Then, one more important thing is whatever molecule we take in making the nanostructures, it has to be FDA approved. When you take a new molecule, it will take you 25 years to come into the market. So what are FDA approved molecules? molecules, like in polymers, these are lactide and co-glycolide structures, which are approved by FDA, and in which, and my lab has been working a lot on cancer and on many other applications. Then it has to be cost effective if it has to go for mass production, and then the easy processing. So today I will be talking about the application of nanostructure to brain. So the main cause of memory loss in the brain is either the traumatic brain injuries, diffusal axonal injuries, neurodegeneration, and abusive drugs. Even if we take the drugs, they also. So the causes of neurodegeneration is the genetic mutations, where we have no way. Genetic mutations also have something with the genes we have ancestors and ancestors. Recently, this from Sweden has been very good about the where he has given ancestors and all that. The inefficiency of cell organelles and is where memories and long time memories are stored and kept. Virus modifies behavior and emotions and prefrontal cortex. So your thoughts of reasoning, planning, etc. Where the memory loss takes place. So the memory loss, and the failure to store memory and the failure to retrieve it is a very tricky portion. The brain is highly plastic in nature. Also, the memory can be lost very easily. There are the drug molecules based on inhibitors and memantine, where we have seen they worked on, they prevent the breakdown of acetylcholine. Whereas these are some of these drugs. The rivastigmine is the one which we have compared our the molecule with this drug, and we have seen that rivastigmine is not working that well. But even the doctors prescribe, because we don't have, approvals of many of these plant-based drugs still in the market. Then we have the memantine-based drug. They have got also the side effects, headache, constipation, and many all. And the drug is Namendra XR, which is being given. And still these type of drugs are given. Targeting. The first will be synthesis, evaluation of targeted polymerasomes for transport of molecules across blood-brain barrier. Evaluation of polymerasomes and postulated with glycosides alone or in combination against amnesic in primary cultures of mouse cortical neurons using molecular tools. In vivo evaluation of the polymerasomes encapsulated with bacosides in mouse model of chemical. As you all know that in India, Ayurveda prescribes as Bacopa monera, and Bacopa capsules are to be given up. They prescribe you 2 capsules in the breakfast and 2 capsules in the evening. But mainly people, when they take these capsules, they always say the pain in the stomach. They are not able to take. But none of them, it is written that they will get the pain in the stomach. So this Bacopa always is a small herb, and it has got a lot of compounds here, bacosides A and B, bacoposides, then sterols, then flavonoids and alkaloids. But our focus was to concentrate on bacosides A and B. And coming to the structure of these bacosides, they are mainly triterpenoids, right? All the 3 are triterpenoids and little bit change. Sometimes we have isopropyl group, sometimes we have isopropene group, and sometimes we have simply the double bond. In the students who are, some of you, if you are from chemistry background, see, in this case, all the glycosides, A glycons are the same. But it is these molecules, this portion, which is always significant for any natural product chemistry. This is the stereochemistry which takes whether the group is axial or if it is equatorial. The whole chemistry changes. That's why when these people, the Ayurveda, they give us these drugs, these Brahmi. I wonder whether they harvest it in the same. Serve the end purpose. So, coming to this, so we Dr. Kritika's PhD was on this BACOP only. So we have demonstrated it has neurogenic potential, neurogenetic properties, acts through tryptophan hydrolase and increases expression of serotonin transporter, increase protein kinase activity, and which plays a key role in learning and memory and helps in the repair of damaged neurons. And although this has been all done where the extract has been taken up, right? And then we worked on another molecule, which is lauric acid, which is one of the components in coconut oil, very small molecule, but doing pretty good job. This is a coconut oil derived compound, medium chain saturated fatty acids, and potential for clearing neuronal hypoglycemia and oxidative stress in AD, PD, and epilepsy. And because of its relative small size, they are easier to encapsulate in polymeric nanoparticles and further enhance their transport across the cell membrane. So as if you know about blood-brain barrier, it's a barrier which is formed by the tight junctions between the endothelial cells in the capillaries. All the students must be knowing that this tight junction imparts BB its impermeable nature. It protects brain, although it protects brain against the trespassing organisms and undesirable molecules and maintains the internal environment in the brain. This barrier also denies entry of drugs for CNS disorders, right? About 98 of the small molecules, drugs are unable to cross blood-brain barrier. So when the doctor prescribes the drug, it is, I think here it is the 2% which reaches, and the rest gets into distributed in our body, and we face nowadays the challenges with the allopathic drugs. So when we started working on bacosides, as it has no ligands, so we encapsulated bacosides in polymerisomes, and there are so many different type of ligands you can attach, like lactoferrin, transferrin, and insulin facilitated receptor mediated transport. And catenized ligands and peptides like albumin can also transfer the blood-brain barrier. But we chose LF, that is lactoferrin, is a single chain iron binding glycoprotein that belongs to the transferrin family. And one of the advantages of lactoferrin to be used as a brain targeting ligand is its low plasma concentration of endogenous LF. So we chose lactoferrin to be, we hooked up to the polymerisomes, which can act as a ligand. And now becomes the role of nanotechnology. As you know, as we spoke, that blood-brain barrier, it is a so tight junctions, and these nanoparticles can be targeted inside and the polymerisomes, and it has got the receptors, that transferrin receptors, which can be taken up by endocytosis, and then we have seen in our work that the damaged neuron gets neuronal survival, right? So coming to the polymeric synthesis of polymerisomes. See, it's a very simple chemistry, not a very difficult chemistry. We have just taken the one TEG of the molecular weight 2000 Dalton, and then with succinic anhydride, the ring opening polymerization. And then we introduced an SS bond. This SS bond is very important. It's very important for brain as well as very important for in case of cancer, because we have glutathione in excess whenever we have any inflammation, and this SS bond, when it breaks, it can release the drug components into the system. We have done a lot of work as anti-cancerous agents using SS bond. And then in this, then again we do another ring opening polymerization being lactide. And earlier I said we have to use FDA controlled, FDA approved molecules, and these lactide and glycolides are all FDA approved. So we played with them. Right? And then we got a structure like this, and then we did another ring opening polymerization with caprolactone. Caprolactone has 5CH2. So this was done to have a lot of drug loading capacity. So we want to have polymerisome which can take lot of drug, because at least 15 to 16% of drug loading has to be there. So in this chain you have SS bond, which SS bond can break up by the glutathione present in the brain. It has an ester link. It can take the pH, it can be degraded, and the drug can come very easily into the brain, right? So this is the nanoparticle characterization and lactoferrin conjugation. You can see now the polymerisome, core and shell morphology over this polymerisome which we made. This is the AFM showing the pictures of polymerisome, and then this is the CD showing, demonstrating the lactoferrin has been conjugated to the polymerisome, and this is then the zeta potential studies where the polymerisome with lactoferrin, the nitrogen content is about 10.72%, whereas without lactoferrin, it is 0.0. We demonstrated that lactoferrin has been conjugated to our polymerisomes. And then we did a side-by-side another study where we synthesize iron nanoparticles, but in comothermal methods, which is available in the literature. And we have one set of experiments where only we have iron nanoparticles loading in polymerisomes with lactoferrin, and another set we have Bacopa with iron nanoparticles and lactoferrin. And these are just some of the studies which we have done. No need to describe them here, but the more important is we did MRI, magnetic resonance imaging, which was done in mass. And you can see the pre-injection, all these hippocampus, platelet cortex, and thalamus. And then we did the study for 60 minutes, 24 hours, and 48 hours. And we could find that in pre-injection, the values of the iron going inside the different parts of brain was very high. And this is the T2 values in various brain organs. And we demonstrated that iron-loaded nanoparticles can go to brain, which we demonstrated by MRI. And this study was done in mice models, right? So after learning from this MRI, we moved to optical in vivo imaging, where we use lactoferrin conjugated Nile red, because for fluorescence, we have to have a fluorescent marker. And here also you can see in the brain, the accumulation was very high of our nanoparticles. Then I'll be talking about in vitro studies on polymerisome encapsulated with bacosides and polymerisome encapsulated with lauric acid to attenuate molecular, cellular, and epigenetic markers in scolopin-induced primary culture of mouse cortical neurons, a comparative study with rivaroxaban, a synthetic drug. So, pardon? Yeah, sure.
So the polymerisome are inserted inside the nanoparticle, or is it a different?
Polymerisome is a hollow vesicle like this, and the drugs are here, and the ligand is here.
That's fine. But what about the iron nanoparticle?
It was inside.
Oh, iron nanoparticle?
Yeah, it was also inside.
So what is the purpose of the iron nanoparticle?
Iron, we wanted to demonstrate whether it is really going to brain. For MRI, you need the iron. Yeah.
Oh, fine.
One crossing blood-brain barrier.
He said he doesn't have vessel cells, they have the receptor for and so the receptor.
So we had N2A cell culture, and we have primary neuronal culture, and now this is BAN is the bacosides, and LAN is the lauric acid now. And these are the nanoparticles. And then we have BAN and LAN in 50-50, 1:1 ratio. And then this is RIN is rivaroxaban. So we had the nanoparticles, all of them loaded separately. And now you can see we did study where we have demonstrated how the neuronal length increases and how the neuronal generation increases. And coming to the second one, so BAN and LAN. This BAN is bacoside, LAN is lauric acid, co-administration promoted differentiation of Th1 positive and 2A cells, which you can see by the relative percentage intensity BAN and LAN. It's very high. And if you look at these pictures, this is lauric acid as such. It does not work that efficient when we make nanoparticles LAN. When we have BAN and LAN together, and you see that the regeneration of the neuronal cells takes place. So Neuroprotection against scolpolamine model. We made a scolpolamine model. Insult was accomplished with neurogenesis, neutrinogenesis, and neutron elongation in BAN and LAN co-administered primary neurons. And look at these pictures, the relative intensity, how they have been changed. Here also it is the length, and here also. What is this, Kritika? Here I'm not able to. Dendrite length, which has increased in case of BAN and LAN. And looking at these pictures, in BAN and LAN, the dendrite length has increased, which is possible only by the nanoparticles, not by if we are taking the Brahmi as such. So this is the upregulation of BDNF and mRNA levels. Now you can see BAN and LAN. The BDNF increase is very high, whereas in case of RI, that is lignostatin, it is not that much. And then mRNA is again very high, which is a positive side of the research. Then we worked on epigenetic modifying properties of BAN and LAN, which was evident by altering DNMT and HDAC expression and enzyme activity in primary neurons. We could see DNMT mRNA, it gets decreased, and then we worked on this 3A also, it gets decreased. Then you can see DNMT activity gets decreased. Then again with the different enzymes, 3M, then 5C5, and HDAC also, it gets decreased. So it means it's decreasing the enzymatic activity which are responsible for neurodegeneration in the mice cells. So in conclusions, BAN and LAN argues neuronal health by remarkable efficacy as compared to FDA-approved drug rivastigmine. BAN remarkably enhances proliferation or neuronal progenitor better than frequently used rivastigmine. Enhanced proliferative effects of BAN and differentiation of LAN could underline the collective neuroenhancement by BAN and LAN combination. LAN showed profound effect on differentiation and growth of neuronal processes rather than cellular modification. So this was the in vitro studies, combination of BAN and LAN and the rivastigmine. Then coming to the in vivo model, that in vivo we could not get lot many mice, and the COVID has started, and pre-COVID also we had lot many issues with the animal house. So we did some work with only bacocytes. So in the gene expression study was done, this was done using the brain mouse in vivo. And you can see BDNF upregulation, CREB upregulation, ARC upregulation. So all these proteins which are responsible for the upregulation we are seeing in the in vivo model also. And looking at the histology, where we have the control, we have scolpolamine model, we have the bacocytes, and we have the BAN, where we can see that this is the normal. And then when we scolpolamine, this decreased. And in BA, it increases, but with BAN, it is much, much better. Histology, if you see, it is much better. They mark the CAI, DG, and CA2. It's much better in case of the BAN. Then the nano formulation of bacocytes efficaciously reverse scolpolamine-induced increase in the expression of chromatin modifying enzymes. So here also in vivo, you can see DNMI, it got significantly decreased. Here also HDAC significantly decreased, and then HDAC C5 decreased. Then again, you can see these different type of enzymes, it gets decreased, and CBP, which is the CRIB modifying proteins, and it gets increased. So we did some study on global 5-methylcystone level post-treatment with BAN in amnesic model brain, and you can see again a very in comparison to scolpolamine, it is decreased, and then the BDNF and ARC promoter methylation state post-treatment with BAN in amnesic mouse model, it gets decreased, right? So in conclusion, the study demonstrates the efficiency of lactoferrin conjugated polymerisomes as the brain targeting agent, which was evident in MRI and memory loss reversal studies in vivo in the chemically induced memory deficient mice model. In this study, we reported bacosides has DNA demethylation and histone acetylation properties in the brain of scopolamine treatment amnesic mice. It was observed that HDAC activity, which was upregulated on scopolamine administration, was rescued by BAN. So to summarize, the polymerasomes based on polycaprolactone and polylactic acid increase the bioavailability of drug molecules by effectively crossing the blood-brain barrier. The study demonstrated the efficacy of lactoferrin conjugated polymerasomes as the brain targeting agent, which was evident by MRI, and the memory loss reversal studies in vivo in the chemically induced memory deficient mice model. These nano platforms can be utilized for the treatment of other diseases like glycomas and other pathologies of the brain like AD, PD, and any other things which essentially required a targeted approach. So these are some of the papers which has been published, and this is the patent also. The workshop was also attended by Dr. Kritika and the conference presentations. And finally, at last, I would like to acknowledge Dr. Kritika Goel for doing a wonderful piece of work. And we had Dr. Ashish Kumar, he was a postdoc from DST, and he also contributed on epigenetic studies. And epigenetic studies was done in Dr. Arpita Konar. She was an Inspire Fellow in IGIB then, and she helped. And the funding we have got all from IIT Delhi, no external funding was asked for. So thank you. I hope I finished on time. You have any questions?
Yes, the seed model
Kritika, please stand up.
what is difference between these two?
So actually we have with Alzheimer's model, so we have taken the transgenic model. So that study we have shown. So whatever we have taken here is the scopolamine model, which causes chemically induced amnesia, and that can be addressed to as transient global amnesia. So it is not specific to Alzheimer's disease, but in most of the studies of Alzheimer's disease, the simplest model which we can take is the scopolamine model. But we have done a specific transgenic study, transgenic animal model studies, where we have shown the lauric acid also in the animals. So that is a different model for Alzheimer's, and in Alzheimer's we have earlier I have also worked on the stem cell model where we are putting amyloid beta directly injecting into the brain, and then we are rescuing it with the help of stem cells. So I would say the specific model would be the amyloid beta model for Alzheimer's disease. So this is just a mimic of Alzheimer's disease model, because it finally creates the memory loss. So we are in this study, we are only checking the memory loss. The behavioral paradigm which we have studied is only about the memory loss. So we are not checking other plaque detection or that studies we haven't done here.
But there also there is a neurodegeneration.
Neurodegeneration is there, yeah.
Not that regenerate.
Yeah, we can rescue it. So we have seen the neurite length is increasing. The number of neurons is also increasing when we are giving in combination. But we have not specifically targeted the plaques, which are a specific symptom of Alzheimer's disease. So we are not talking about the plaques here. In another study, we have taken transgenic animals.
So is the time of administration important in respect to the degree of neurodegeneration which has taken? Once the plaque is made, then I don't think that.
In this is the plaque study is not this one. So we have this transgenic animal. So they already have the plaque. So in that, I think it's not about the time. So when the time which we're talking about, so that would be when we do through stereotaxis, we have done one model. Through stereotaxis, we inject beta amyloid. So that time it will we have to optimize the time, how much time it is taking for the plaque to settle and come as a callus model. So in that case, time is important.
Any further questions?
Thank you very much.
You asked what you asked.
No, I request Sanju to please felicitate.
So I'm pleased to welcome the next speaker of the day, Professor Rana Pratap Singh. He is currently affiliated to School of Life Sciences, Jawaharlal Nehru University. He has a PhD in cancer biology from JNU. He has received a postdoctoral training award for prostate cancer research from the Department of Defense, USA. He has also received ICMR International Fellowship for Young Biomedical Scientists to visit University of California. He was assistant professor at the Department of Pharmaceutical Sciences, University of Colorado. He has received Indo-US Science Technology Forum 2019 award for establishing Center for Integrative Cancer Biology and Therapeutics. He is also worked as professor at the School of Life Sciences in the Central University of Gujarat. His areas of research interest are carcinogenesis, cancer chemoprevention and therapeutics, chemoangiogenesis, radiation therapy, anti-cancer drug development from natural agents, steroids, and cancer stem cells. I welcome you, sir, to kindly deliver your talk.
Thank you very much for the introduction to both the chairs. I'm also very much thankful to the organizer, especially Professor Alok Bharti, for giving me this opportunity to discuss our work. So what I'm going to talk about is just a small work done by a PhD student in my lab. And she was working on radiation therapy. And also she wanted to know that if there is any compound, whether it is going to have any radiosensitizing effect or not. And if it is, then what could be the possible mechanism? So I will focus on that aspect. And for that, she took the example of prostate cancer. Since we work in several cancer models, so for this, prostate cancer was selected. You already know about the disease progression with respect to cancer. Starts from normal prostate epithelial cell, then thin adenocarcinoma. Usually when this prostate cancer is diagnosed, the therapeutic options that we have, you can see here androgen or androgen receptor targeting therapies. But what happens after a few years, the prostate cancer regrows. And at that time, those cells will become androgen independent. It means they are not responding to the therapy, or they will have autocrine growth factor receptor signaling, and at the same time, deregulated cell cycle progressions. And there are some other mechanisms which have been observed. But at this time, the intervention what clinicians are going to do for the patient is either radiation therapy or chemotherapy. So here, with respect to radiotherapy, there are some challenges. Development of resistance is one, and the toxicity is another one. These are two major challenges, and they depend upon the sensitivity of the tumor tissue as well as normal tissue. And therefore, the successful outcome depends what kind of cancer cells and what kind of dose is being used and how the patient is responding to the treatment. So for the study, we took one simple compound. This is silibinin, flavonoid compound, on which we have worked a lot. This is just, it's a structure showing hydrophilic and lipophilic portion of the molecule, suggesting that this molecule, if it enters into the cell, it can have a range of interaction, starting from extreme hydrophilic to lipophilic. So we did one experiment. This is a thymic mouse model. The advantage of this is that you can grow human tumors by using human cancer cell lines. So what you see here on left hand side, you have control group. The tumors, you can see they are bigger as compared to the other side, in which the siliburin was given in diet, and this treatment went on for 8 weeks. It was only 0.5% weight by weight. So this experiment suggested that this compound may have anti-tumor effect against prostate cancer. And next, we wanted to know what could be the possible target of this compound. We did one simple experiment. This is clonogenic assay. You can see the doses 2, 5, 7.5, and 10 micromolar of siliburin. This is 9L rat glioma cell line, which does not express epidermal growth factor receptor. And we expressed human EGFR in this cell line. If you look at all these wells, this drug is not showing any effect. The number of colonies, you can see it is same. So once the EGFR was expressed here in this 9L rat glioma cell line, now you can see the strong effect of this compound when the dose is increasing. And this A431, this is skin squamous cell carcinoma cell line, which is known for high expression of EGF receptor. This is also showing almost similar trend. Then we thought that most likely this compound might be working through EGF receptor. And you might be also knowing that EGF receptor is kind of deregulated in almost 50% of cancer cells, cancer types as well. If you go for it's a signaling pattern, this is a classical activation of epidermal growth factor receptor through ligand mediated. You have EGF, TGF alpha, and amphoreglin. However, you might be also knowing about ligand independent activation of this receptor. This is stress induced activation, where even ultraviolet radiation, ionizing radiation, or some other stresses can also activate this receptor to transduce mitogenic and survival signaling. We came across a paper in 2001 where it was first time reported that EGF receptor, this is a very big molecule, 170 kilodalton. So this molecule was found to be in the nucleus. Then the scientists, they got surprised how a big molecule, which is supposed to work on the cell membrane to transmit the signaling, why it is going into the nucleus. At that time, we realized that if such a molecule is entering into the nucleus and going to work as a transcription factor, the whole concept of signaling is going to change. However, later on it was found that EGF receptor is not having any DNA domain. So it was ruled out that this is going to work as a transcription factor. It means if it is going into the nucleus, then it has to have some functions. So that was the puzzle when people started working on. So on left hand side, this is the classical signaling of EGF receptor, and it can influence almost all the processes of cancer development: tumorigenesis, proliferation, metastasis, chemoresistance, or radioresistance. On the right hand side, if you look at nuclear EGF receptor, we focus on this one, that this receptor is coming into the nucleus, and what could be the possible outcome. Then we focus on the last one, whether this is having any role in radioresistance or not. With this came, we looked at the literature and found out that in clinical samples as well, if the patients are going under chemotherapy or radiotherapy, the presence of EGF receptors in the nucleus as well. However, there were no reports in prostate cancer. Then we did some simple experiment here. We did prostate cancer cell lines exposed to the ionizing radiation. And first we did clonogenic assay, D145 advanced prostate cancer cell line, then PC3. You can see the dark line. This dark line is for only radiation treatment, and this less darker one is radiation plus siliburin. In both the cases, this less dark line is below, indicating that colony formation efficacy is getting decreased in the presence of siliburin. This is another cell line from lung cancer. Just we wanted to see whether it is there only for prostate cancer or also for other cancer. Similar result was there also for lung cancer cell line, A549. The more interesting observation was here, human embryonic kidney cell line, which is considered as non-neoplastic, non-cancerous. And here the result was exactly opposite. You can see that dark line is below, and with the radiation plus drug combination, it is above, showing that in the normal cell line, this drug is, this compound is having a protective effect. So here we got it clear that there is a selectivity. Nowadays, this is what everybody is looking for, an anti-cancer drug, how to make it selective, cancer cell versus normal cell. And that is what we observed here with this compound. Then we did some experiments related to cell cycle progression. And this you will find that in the combination, that is black bar, in all the cases, GTM phase arrest was there. In these cell lines, some molecules which are associated with GTM arrest, cyclin B1, CDC2, CDC25C, all the molecules changed in that favor for arresting GTM phase. So that is the summary of this, how CDK cyclin B1 complex, if you inhibit, you will get GTM phase arrest. And that was happening with this combination, ionizing radiation and silibenin. And this is a very important checkpoint over here. If you block it here, cells are not going to enter into the mitosis or the cell division phase. Then for the selectivity, we just had to look at reactive oxygen formation. What is the status in this cell line? And here again, we got very interesting result. You just look at the last figure, figure D. First bar, control. The next one is only silibenin. Third one is only radiation. Fourth one is the combination of radiation and the drug. You see, the reactive oxygen species in case of cancer cell line, that is G145, ROS level is increasing in combination. But when you go to the normal cell line, the result is very drastic. You don't see the increase in ROS, but ROS is being decreased in the combination. Another important observation here we found the third bar, if you look at the radiation treatment, the third bar is radiation treatment. In both the cases, the bar height is same, whether it is cancer cell or normal cell. So this tells that radiation does not discriminate between cancer cell or normal cell. It's same in both the cases. However, the combination of drug is differentiating, and that could be a possible mechanism for protection of the normal cells.
Both these cell lines expressing EGF, same level?
Same level I cannot say, but EGFR is there. Expression is there. So some more experiments were done related to apoptosis or cell death, and we find more cell death in the combination as well. This is related to checkpoint. So checkpoint inhibition was also observed. You see check 1, check 2 phosphorylation in the last treatment, that is very much reduced. Now I will come to the main point of the study where we are looking for EGF receptor, how the drug is modulating in the translocation of EGF receptor. This is G145 cell, cancer cell line. The blue color is nucleus, Doppler staining. Red color is for EGFR, this is merged. If you look at very hot treatment, still the red color is outside of the nucleus. There is no overlapping of the red and blue color. But when the cells are exposed to radiation, now you can see all the red color, it is coming into the nucleus. And also overlapping of blue and red. So this tells directly that EGF receptor is getting translocated into the nucleus once you expose the cells with the radiation. When we combine the radiation with the drug, then you see it's very clear nucleus. Again, red color is outside of the nucleus. So here you can conclude that the drug is blocking the translocation of EGF receptor into the nucleus. So those are the conclusions. Just to support that, we have enlarged image with the radiation, EGFR in the nucleus, radiation plus silibenin, EGFR outside of the nucleus. Again, you need to substantiate your results by doing Western, make fractions, cytosolic and nuclear, and show that where EGFR is located. So you can see here in the radiation 5 gray, in the cytoplasmic fraction you don't have, here in the nucleus you have, but when you combine, then you have opposite result. Here in the nucleus, you don't have EGFR localization. Now next, since our hypothesis was related to DNA repair, we also thought to look at DNA PK, DNA dependent protein kinase, which is very important for DNA repair. Here red color will represent the active form of DNA PK, that is serine 2056 phosphorylation. Green is again nucleus. Control and silibinin treatment alone, you will find similar observation. With the radiation, again you see red color is going into the nucleus. This tells that when you damage the DNA with the radiation, the DNA PK is entering into the nucleus to repair the DNA. And that is the mechanism how it usually happens in all the cells. But when we combine the drug with the radiation, again you can see the green color, it's very green. There is no overlapping. So this tells that the drug is also blocking the nuclear translocation of DNA PK. In other words, if you combine both the agents, EGFR as well as DNA PK, the translocation was blocked by the drug which was being induced by the radiation. Now, this is again very important data related to the DNA damage in the form of S2X foci you can measure. If you look at this bar diagram, the black one is 6 hours and next the less darker one is 12 hours. So whenever DNA damage happens, immediately DNA repair machinery will start in the cell, they will start repairing the DNA. And the kinetics of DNA repair in the treatment versus non-treatment, how it is happening. So if you look at this 6 hours versus 12 hours, in the 5-day, there is a decrease of DNA foci. It means most of the DNA damage was repaired. In the combination, 5-day plus 7, you can see this decrease is very less as compared to this one. So in 6 hours, the amount of DNA repaired in the drug treatment after UV exposure is very less. So in other words, in the cancer cells, when the drug is there, the DNA repair might be getting impaired, it is not efficient. And when the DNA damage is there, then likely what we expect, the cells are going for death or apoptosis. Next question you ask, let me just summarize this, how it might be happening. So EGF receptor and DNA PK with only ionizing radiation, they are entering into the nucleus, and wherever DNA damage or stain breaks are there, they are going to repair through Ku70, Ku80. There is a very well-defined mechanism. But when we combine radiation plus silibinin, both the molecules, they are not coming into the nucleus, then there will be deficient repair, and corresponding we are expecting that there will be more and more cell death. The next question will come whether it is the similar in the normal cells or it is also happening the same mechanism what is there in the cancer cells. It means where this mechanism is different in cancer versus normal, it is same. So we took keratinocytes here of the normal cell, did the same experiment. Here for EGFR you can see the red color, again it is in the nucleus. In the normal cell, it tells that DNA repair mechanism is there and EGFR might be playing a role. But when we combine it with the radiation, here is still EGFR is in the nucleus. However, this result was very different in the cancer cell. So there is no blockade. So this tells that if DNA damage is there in the normal cell, this mechanism will operate for the DNA repair. And that could be again a protective mechanism for the normal cell. So we just try to do one in vivo study to see whether similar things are happening or not in vivo condition. This is vaccinated doses as the patients are treated in the clinic. Total 15 gray dose we gave, and the same cells were xenografted. If you look at the kinetics of tumor growth here, tumor volume, blue line is for control, tumor is growing. And you just focus on this pink line, that's it, radiation plus the drug. You can see this line is going down, indicating that tumor is regressing in the combination. So very strong effect. This is at the end tumor weight. It is again very significant effect. In terms of systemic toxicity, body weight, as you already know that radiation has a very strong side effect. So this red line indicating the body weight loss after radiation treatment up to 15 gray. The pink line, which is combination, it is showing recovery in the body weight loss. Here also diet consumption is getting recovered. Another important parameter is immunotoxicity. When radiation is given to the patients, immune toxicity is a major concern. Again, if you look at the third bar in all the cases, ionizing radiation, total WBC count, lymphocyte count, platelet counts, they are getting decreased. But in the last bar where combination is there, everything is coming back to the control. So immune protection is also there in the combination. We also did some immunohistochemistry just to look at the tumors, what is happening. This is related to proliferation. This is related to S2X, DNA damage, or apoptosis. All these results were in the favor of the combination. So now I'll just summarize whatever I have discussed. EGF receptor, it is on the cell membrane. This is its classical mechanism how it transmits signaling for growth and proliferation of cells. What we observe here that EGF receptor is getting internalized into the nucleus, and in the nucleus it has role for enhanced DNA repair. On one hand, we are trying to damage the DNA in the cancer cell to kill them, but this is a resistance mechanism. EGF receptor is coming and trying to repair the DNA, and the cell will survive. It was again supported by the translocation of DNA PK, that yes, both are coming to the nucleus and DNA repair will happen in the cancer cells and they are not going to die. This is a resistance mechanism. And what this drug is doing, once we put the silibinin along with radiation, it blocked the nuclear translocation of both these receptors as well as DNA PK, and then these cells become radiosensitized, and that is how we got more and more cell death in the cancer cells. So this is my group in the laboratory. There are students who are working on different projects. However, for this project, as I said in the beginning, this was a PhD work of one of my students, Dhane Nambiar. And for this work, she also got Fulbright fellowship. She went to University of Colorado, did some work over there. She was also selected for this Nobel meeting in Germany, and then went for postdoc. Currently, she's working as instructor in the radiation oncology department in Stanford. Funding was given by DST and UGC. So this, thank you all for listening. And if there is any question, I'll be happy to answer.
Radio resistant cell line.
LNCAP, PC3, and G145. So first we did the experiment to find out which one is more radioresistant. So G145 is the strongest in this term. It's a highly radioresistant cell line, and that is how it was used.
Professor Anand,
Sir,
it's a good piece of work. A couple of questions.
sir.
I think first concept that nuclear translocation of EGFR by I think Min Chang, the Chinese guy from MD Anderson, published bunch of paper including cancer cell, etc. Started around 2000. But my question to you, using your silibinin and response to that radio resistance, most important point when there is a settling of EGFR, what is BARTK, receptor tyrosine kinase? What is the status of tyrosine phosphorylation of EGFR when it is translocating into the nucleus?
So usually, sir, there are a lot of studies on that, and we are also working on that. So I'll just show you. I have one slide for that. So this is a very particular phosphorylation, 30654.
Some tyrosine residue also gets phosphorylated.
So this is known. This is already just published that if 30654 is phosphorylated, then this will help in settling of this receptor into the nucleus. And there are also some phosphorylations which are working as a negative.
Okay.
So both positive and negative, but still people are working on to find out. There will be many more.
Second quick question. You know, when you think about that tumor microenvironment in a real scenario, for example, patients who are developing cancer, in that microenvironment, not only tumor cells, there are many stromal derived cells, stromal compartment, they play a very important role. And the expression of EGFR also reasonably high. For example, CAF, cancer-associated fibroblast, maybe some of the immune cells, including macrophages. Have you looked into those parameters in your preclinical animal model or in the patient's tissue, cancer sample?
Yes, some of those experiments have been done. However, here it is very peculiar. One cell versus another cell. You are saying CAF and cancer cells. So definitely they are going to modulate the activity of cancer cell. But here what I am talking, within the same cell, if there is a receptor on the membrane, how it is behaving with the radiation treatment and the drug treatment. Because I don't think that EGF receptor from CAF is going to enter into the cancer cell. That may not come. However, that interaction may be possible. Yeah, so that interaction may be possible, but it is unlikely that it is going to enter from one cell to another cell. So, thank you, sir.
Is you use the cerebellum. Did you change the percentage of cerebellum or you kept it constant?
It was constant in all the experiments.
And then
Because all the experiments were done to find out a particular dose
okay, on
that
that
we
basis.
export. Yes.
And it's a pure compound.
Pure compound.
Then in which plant is it present?
It is called as milk thistle plant. Milk thistle. And you might be knowing about artichoke. It is used also as a vegetable. So in artichoke also it is found up to 2% weight by weight. So that is quite high amount. And
Does cerebellum also make some bring over some difference to phosphorylation?
EGF receptor, yes, it inhibits. It inhibits.
So what would be the possible reason why this catheter cerebellum treatment blocks the entry of the EGFR into the nucleus? Like, is it because of the change alteration in this?
And that we are still working on that. Like there are several possibilities. One, as you said, phosphorylation. There may be some phosphorylation which may be inhibitory in the nature, and particularly cerebellum might be doing that. And the second, interaction with the other molecules. And third, it could be the modulation of the nuclear pore. It has to enter through the nuclear pore. And there is a very complex mechanism how proteins enter into the nuclear pore and then come into the nucleus. So drug may work like that as well. So these are the possibilities to explore.
So are there, other than EGFR, are there any which can get altered with it?
There might be many. There might be many. Thank you.
Thank you for Professor Anju to felicitate Professor Singh.
That's what
I would like to speaker, Professor Smita Singh Pawar, who is the head of Department of Genetics, Osmania University, Hyderabad. Professor Pawar earned her PhD from Osmania University, Hyderabad. She is also the recipient of Young Scientist Award by BMMRC, as well as a DST Broadcast Fellowship. Professor Smita is also an excellent teacher, as she is a recipient of a State Teacher Award. I would like to request Professor Smita to proceed. Presentation.
And thank you for that excellent introduction.
Also, let me thank the organizers for having me here.
It's wonderful being part of this great conference. Secondly, after such experience, people have talked here, so I'm a bit shaky. They could have so much experience behind me. So I'll try not to get on the tempo what they build up. So this is, as rightfully, as introduced. So this is Professor Smita Pawar, Head Department of Genetics and Biotechnology from Osmania University. So before I could start this topic, I'll just try to briefly describe how do I fit into this. I'm basically a cancer biologist, which working on genomics part of it. And why cervical cancer? Cervical cancer, when I started, I wanted to work in cancer biology, but when I was asking myself, like, what cancer should I be working on? Why did I pick up this? Very first reason, if you see, breast cancer happens to be the most common cancer, and then I think CC is coming up in a very big way, and I think it is not very long that it's going to take over cervical cancer. Secondly, it gives you a very broad window for you to work with. There are many cancers that you really do not have the chance to help the patient, but here, one thing that is possible is first, by identifying it at a very early stage, we can reverse it. Secondly, it gives you a lot of time for you to treat the patient. Almost you get a window of 10 years, which most of the cancers do not give you. So these are the two reasons why I started with this. And then how did I start, this work start off? We have done this genomic study where the data regarding Indian population in specific is very, very scarce. So we did a very small pilot study with the South Indian, especially from one cancer hospital, that is the MNG Cancer Hospital in Hyderabad, which caters to a whole of the Telangana. So the whole state's cancer patients who are like below the poverty line are sent to this hospital because that is the only hospital that caters to it. So when I've seen the scenario where till date, I think we're having 2 or 3 samples that I've got which are HPV negative, but most of them are HPV positive. So I can say that from my sample, 99% of the samples are HPV positive. That was the first time, and then the data regarding the population, because the socio-economic status also plays a very crucial role in cervical cancer. So I just wanted to see how it is it really different? No, it's basically it's not supposed to be different, but yes, what is the finding that I get from my study? From the transcriptome data that I got, the summary of it, I could say that the genes are the same, which are playing the role. But yes, the ranking is changing. The most prominent gene in the Caucasian population is not seen in us. And the ones which are playing prominent role here are not really figuring in the top 10 or the top 20 or top 100 genes if you're going to take. So that is how the whole study started off with. Then we identified certain genes which were playing very crucial role, but were not identified by the other populations, definitely not in cervical cancer. One such gene was osteopoietin. Gopal Pandit was an expert in this field. And then we started looking at what are the other ways and methods of stopping this gene or inhibiting this gene. The expression was not only seen in the cancer patient, but also in the recurrent cases. We had done a transcriptome analysis for the recurrent cases where this gene was constant in both the population. Though the sample size was not really huge, but it showed that even for the recurrence, it was playing a very major role. So we had picked up that gene, and then we started looking for, are there any known analogs? Rather than starting to develop a new molecule which is not our expertise, we wanted to see which is already available. And andrography happened to be one of the biggest hits, which shows a really good compatibility in inhibiting in silico with SPP1. So here, secondary metabolites are adjuvants for cervical cancer, a multi-target therapy. So the whole idea of this project is, cisplatin happens to be the only drug that has been administered to the patients who are coming to this hospital. The resistance definitely is there. So we have conceptualized this idea because some of the genes that have come up. So if we can add something with cisplatin, which can make it more effective. It's not like replacing it, it's something which will make it more effective. So we started with this idea. So as I just generally, there is the general introduction, I think, which I will skip here. So here, cisplatin is the drug treatment used as chemotherapeutic agent for many years and in the later stages of the cancer. As I've said you, one thing is HPV positivity, and secondly, most of the samples that we are getting are only in the stage 2A, 2B or 3. That is on the first stage we have higher number of samples to work with. So I'll come to this picture once I finish with the whole thing. Yeah. So we started with the set of genes that we identified in the study, where PI3K was the biggest hit, NF-kB, COX-2, and PTEN. So the same data we had to analyze, but our sample size was very less. We have analyzed it across the other databases that were available, and yes, it was showing the same kind of a pattern across various populations. Now, the same thing we have reconfirmed it with histochemical analysis, IHC. These are the patient samples. If you look at it, the PI3K, you have a very strong positive. Then the same thing with the NF-kappa B, we see a very strong positivity here when compared to the controls. The COX also has a very strong cytoplasmic presence, and the PTEN, we haven't found any PTEN in this. The data was further analyzed by doing the qPCR analysis, where the similar results were seen. Further evaluated the rock curves. And these are the some, I'm not put most of the pictures here, just a representative picture where we have done the molecular docking, and these docking studies have been done at VIT where Dr. Anthony is our collaborator who does all the in silico studies, and he gives us the list of the molecules. So androgen was one of the biggest hits that was proposed by the VITR. When we validated this, whether, there were already a lot of literature that was present, still we had to redo this thing again on HeLa and SeHA cells, and here we validated the inhibitory effect of androgen on the cervical cancer cells. As you see, it does show good inhibition here, and this paper was published in 2021. So this is the same thing showing the inhibitory effect of the androgen. We performed certain assays as a cancer biologist, so we are all aware of the transfer migration assay. We are just checking whether the androgen is able to inhibit this migration. When you see that the HeLa is treated with androgen, there's a gradual reduction in the migration as well as in the SeHA cells. Then the nitrogen inversion assays, as the concentration is increased from 5 micromolar to 10 micromolar, we find that the migration of the cells is drastically decreased, both in HeLa as well as the SeHA cells. We are looking at what are the targets that this androgen is hitting. As I told you, PI3K was the biggest hit on the transcriptome data that we have generated from the patient samples. So we started with the PI3K. And here you can see that the PI3K after the treatment, gradually you can see a decrease, a substantial decrease in the PI3K. And then PITT also was gradually decreased, but no difference was observed with the AKT. The same kind of results we are seeing with the SeHA cells. The similar experiments were performed with the NF-kappa B, COX-2 and PTEN. We find substantial increase of the PTEN with the treatment of androgen, and the COX-2 and the NF-kB, we see it gradually going down. The same results we have seen both in HeLa as well as in SeHA cells. We looked at some of the apoptotic markers. The BAX has gone up and the BCL has gradually come down. The similar results again are repeated in the SeHA cells. We did an ICC assay to see the NF-kappa B. So there's nothing when compared to the slides that I have just seen. So here also what we see is the NF-kappa B is gradually being decreased with the treatment of the androgen. So we tried to replicate these experiments in vivo. This was a bit of a difficult task for us because maybe the model that I used was not an appropriate model. But because we started with C57BL6, we had taken these mice, but initially when we tried to grow the tumor with the HeLa cells, and we couldn't do it. So then we had to immunocompress, suppress the mice, and then we had to go ahead with the treatment. That gave us certain, the results were seen, but I think this is the protocol that we have standardized, and I think it's a standard protocol done in most of the labs. If you look at the tumor size, when compared to the control, the treatment has shown gradual decrease in the tumor size. The IITs are performed with activities that are obtained from the army. So in the first one, you will find that the PI3K, the level is mild to no IIT that has been seen. MF kappa B is also mild to moderate, and the COX-2 levels also have decreased to mild to moderate. And P10 level has predominantly seen in the nucleus and from no reactivity to moderate level. So this is in sync with what all data that I have shown in vitro. So in vivo also angiograph was showing the same kind of an impact. So this is what the pattern that first escaped. So here angiograph is not only targeting one, but many hits are there. So it is very effectively inhibiting the PI3K, MF kappa B, COX-2, and at the same time it is initiating or there's an activation of the apoptotic markers. So this is what we are proposing is a combination therapy of for treatment of cervical cancer, that is angiograph in combination with cisplatin. So we are now developing cisplatin resistant lines to see whether angiograph can make any difference to that. So that is still the study, they're still going on, and this project has been funded by CERN. So thank you, and I'll be ready to take some questions.
Mr. Smita, it's a nice talk.
Thank you,
So
sir.
just quick 2-3 question and comments.
Yes, sir.
Is angiography working? HPV positive cervical cancer or HPV negative
So
cervical?
like I was from HPV positive, so negativity.
Second question.
Yeah.
When you talk about PI3 kinase, it has two subunits. One is catalytic and the regulatory.
Yes, sir.
P110 and P85.
Yes, sir.
You have shown P85, expression of P85 going down.
Yes, sir.
You should check the level of phosphorylation of P110 and P85.
Sure, sir.
Third question, you shown that NF kappa B. NF kappa B main subunit is P65.
Okay.
Whether heterodimers or homodimers along with P50 or P65, P65. You should check the level of phosphorylation of P65 rather than seeing only NF kappa B.
Sure, sir.
And what is going on is endographylite enhancing or inhibiting P10 expression. What is going on?
So
Or
P10
phosphorylation
expression,
P10.
so right now we have the P10 expression and it is showing increase in the expression of the P10 levels, both in vitro and in vivo.
Because being a chemist suppressor, maybe we should have a combination of P10 along with endographylite. That might be other than that other chemotherapeutic drugs what you are talking.
Yes. Thanks for this suggestion, sir. Definitely we'll try to incorporate because still this is an ongoing project.
Thank you.
Any other questions? Thank you. I hope again. I hope again request Professor Angie to.
So our next speaker for the day is Professor Arun S. Karat. He is affiliated to School of Life Sciences, Jawaharlal Nehru University. He has his PhD from Indian Institute of Sciences, Bangalore. He did his postdoctoral research from Rockefeller University, USA, and Université Libre Arts, France. He worked as a scientific assistant at the Department of Biological Sciences, TIFR. He was the academic director at Dr. Babasaheb Ambedkar Marathwada University. He was the director of UGC Human Resource Development Center at the same university. He is the director and professor at Dr. Babasaheb Ambedkar Marathwada University. And currently, he is working at the Jawaharlal Nehru University as professor. I welcome you, sir, to President.
Good afternoon to all. Our group works on antimicrobial and anti-cancers, especially from the medicinal plants and the beta-lactams inhibitors. So today I'm going to speak one of the aspects that we deal, that green synthesis of cellular nanoparticles, which are doped with the phytochemicals and their potentiality as an anti-cancer. So as we all know, this is about the size, and there are like examples of biomolecules which are in the size range of the 1 nanometer to 100 nanometer that covers a variety of liposome, dendrimer, gold nanoshell, quantum dot, fullerene, and that covers basically the glucose all the way up to the bacteriophages and certain viruses also. So there are various metal nanoparticles, or metals have been considered as a material for reduction into production of nanoparticles. However, the gold and silver have assumed quite a good position due to the fineness in the structural form and the dispersion of the nanoparticle per se. Otherwise, you can see like copper is there, iron is there, many of the divalent cations as well as trace elements have been considered. And they also have different kind of physical properties as a metal and as a nanoparticle. There are different physical and the chemical properties. So the size spectrum, if you look into that, it starts with the 2 angstrom, that is a water molecule, and all the way the protozoans, which comes to the 2 micron, to the viruses, that is where the biological range of the certain molecules. When we talk about the activity, bioavailability, it basically covers the bioaccessibility, absorption, and transformation, multiplications of each. Now, bioavailability talks about the material to be made available, solubilized form, and its liberation to be able to make an effect to the site of action. So absorption basically has a transfer across the mucous layer and epithelial cells, and transformation basically to bring about the reaction so that the chemical and biochemical reactions would result into the favorable or unfavorable interactions. So there are 2 approaches, top-down and bottom-up, and as we know that the top-down would break the metal piece into the small molecules, finally into the exfoliation as a multi-layers of monolayers or into the nanoparticle. And the bottom-up approach will start with the precursor, and all the way it will produce the condensed matter that is into the nanoparticle, and they could have a different type of structures, diamond, graphene, nanospheres, nanosheets, the nanowires, the fullerenes, etc. are made available. Number of nanometers we are very well aware of. So when we talk about metallic synthesis, here, especially in the green synthesis part, includes the biological material. Rather, we can have a physical as well as a chemical approach for the synthesis of nanoparticle. So plants and microbes have been so far tested, as well as recently the insects have also been used as a material for synthesizing the nanoparticle. And we could consider leaves, roots, flower, fruits, etc. are a part of the plants, or the whole microorganism, a bacterium, a fungus, or the algae extract per se as a material. That would be the brain of it as we use yeast extract in our laboratory media preparation. So this is an approach where you have to collect the plant material and make into the small pieces, of course, by surface sterilization and washings properly. And then heat as a principle, you can mix it with the particular silver or gold. Here we use a silver nitrate, and then you'll get a change in the color which indicates there is a property change, and there you can characterize the matter in that changed color, and that's where you can know whether nanoparticles are synthesized and what type of nanoparticle structurally and then to the functional part. So basically the extract contains all sort of secondary and primary metabolites. You can have proteins, you can have amino acids, you can have oligopeptides, you can have alkaloids, you can have flavonoids, you can have terpenoids, vitamins, sugars, and lipids also, and certain popular enzymes, which themselves work as oxidative reductase class of the enzymes. So that all material is available. So one has to figure it out what exactly is participating in the process of reduction and what is written on the nanoparticle. Otherwise, synthesis of nanoparticle with physical, chemical, or biological means at par same. However, use of brain synthesis approach is meant to reduce the energy and the thermodynamics required for A, and the second, the efficiency. Because often a natural compound that we know, a few of them have been known, J7 beta-acylidimine, and there are at least 9 different chemotherapeutics used in cancer are actually plant-based. But we still try to find more of the phytochemicals and more of the natural compounds because they will have more bioabsorbability and bioavailability. Therefore, with reduced side effects, because they are used in the cancer therapeutic, one of the biggest downside is the side effects. And therefore, the looking forward for the natural product is the business currently ongoing, and therefore, there is a huge amount of scope. And the nanoparticle, particularly the themselves are this small, they are the delivery vehicle molecule. They will selectively create the band of phytochemical, which is really and truly very small in size. So, as of now, the molecules we know by the different mechanisms. Here also yet another way to know which are other small molecules or the tiny, very micro nano sized molecules. We can figure it out. Those will bring an impact or make an impact onto the anti-cancer properties. So, types of nanoparticle has been known. A goal has been very safe because the smallest nanoparticle, the finest nanoparticles so far known are from the gold up to 5 nanometer, and the mostly spherical, or if you can play around with the ratios and the temperature, the protocols, you can change the type of the size and the quality of the nanoparticle with the gold as well as with the silver. So the mechanism of synthesis, as I mentioned, that there is a blend of phytochemicals present in the extract, and that would interact with the metal ion, and then it would result into the production of, basically the reduction results into the production of the nanoparticle. So it has already been developed to have several sort of applications, includes the various types of cancers, brain, skin, lung, breast cancer, prostate, colorectal, bladder, and for imaging cosmetics. There are large number of cosmetics available from over 4 decades that contain the nanoparticle. Before even we know it, nanodialect technology does have a big potential, and we have been using lots of lotions that contains already the nanoparticles. So biomedical application, catalytic applications, food industries, everywhere you can see it has been used as a potential, and one of the aspects that I already told that we also work as a nanoparticle for antimicrobial potentiality. So when we talk about the prevention of bacteria, basically it either destroys the bacterium per se or targets one of the cellular, subcellular organelle, so that the bacterial replication or survival is jeopardized. Ultrasensitive detection has also been made as a sensor material. Then pathogen removal from the blood, that is septicemia particularly in that case, or you can say like parasitemia also, there also you can have an application of it. But here particularly I will be focusing more today about the advanced drug delivery, because nanomaterials and its application in terms of the natural products to deliver, because every metal ion if we use has its own localization, and thereby the phytochemicals will be easily traveled all the way with this nanoparticle to that site of action, and there you could be able to get its reaction as effectively as possible. For example, aluminums are known to disrupt the cell walls. Bismuth is known to alter and modify the process of Krebs cycle, that is a bioinertics. Particularly in the case of gold, nanoparticle, gold is known to disrupt the cell wall, as well as a DNA inhibits, it intercalates between the DNA like bacterial virus does, and DNA replication and transcription is prohibited. Silver nanoparticle, on the other hand, rather than interconnecting, it affects the topology of the DNA. So rather than sitting into it, it basically affects the structural organization of the DNA and prevent therefore the DNA replication process. So in all the cases where nanoparticle or the single nanoparticle, what is expected is replication is going to be prevented. And in addition to that, electron transport chain, as like in the case of bismuth, silver nanoparticle has an advantage because that's mostly important process in the biology where the ATP synthesis and ATP utilization is carried out. So energy transfer and the DNA basically duplication is going to be affected. So choice of silver makes justifiable. So one of the, I will be talking about two different plants. The first plant is Solanum xanthocarpon. As by the photo, you could see that it is available everywhere, all parts of India, especially the dry area or the semi-arid area. It is quite rich with this plant. So when we synthesize the nanoparticle, we checked with the 10, we can say that around 22 or 20 nanometer was the average size, which again fits into the standard size so far documented for the silver nanoparticle. 10 is the smallest and up to 30 nanometers is considered to be the good quality silver nanoparticles are. There was spherical monodispersion that was seen when XRD is basically to support that the dispersion and there is no monodispersion and there is no agglomerization or culmination of the different nanoparticle. Septor also showed that there was quite a few OSCH, then NHCF, carbon-carbon double bond, carbon-carbon single bond, like functional groups are available which have known impact on the biochemical rate of reactions. Here the whole fruit extract, then whole fruit extract as a reducing agent and nanoparticles were synthesized. Then nanoparticles were either with aspartate or with the arginine. And the chemically synthesized nanoparticle, that is basically the sodium citrate as a reducing agent and prepared. So that's a very standard one which is known. So all the way up to 800 microgram per ml, the concentrations were used, but it is very clear that the first two, if you can look into the 600 and 800 nanometer, particularly here, the whole fruit extract has a more inhibitory activity, and the second comes the nanoparticle. But when you have a nanoparticle coated with the aspartate or coated with the arginine, which is a very well documented biofunctionalizing amino acids in the literature, they miserably failed. So it's not a rule of thumb that something is published somewhere, it is written, and it would always go the same way. So this is where we could confirm that the extract seems to be far better, even better than the nanoparticle. So it's not that every plant you take it and you will be synthesizing a nanoparticle and making it as anti-cancer. That's also not true. So this was a message wherein the nanoparticle can be produced only from the fruit extract, whereas we tried from the roots, we tried from the leaves, we tried from the stem. That part either did not have activity there itself, or nanoparticles were also having no activity. So this is only the part where we could see the activity. So the biochromatography was carried out initially too, because I told my student not just to document the nanoparticle based activity, just go further down and go more into detail and try to figure it out what particularly the phytochemical it is there. Otherwise, what normally somebody will do is process the extract to know what sort of phytochemicals are present. That will give you qualitative or quantitative, but it will never tell you what is a reducing agent and what is doped on the nanoparticle. So that was something she had to do it. So column chromatography, 64th and 65th eluate did have the absorbance around 420 nanometer. That resembles to the silver nanoparticle. So she processed the 64th eluate further for the synthesis of nanoparticle with that. And then the activity was checked, obviously that did retain activity. But again, the eluate is not a fine purified compound. It may have more than one compounds or a blend of compounds. It is something better than the original crude extract. So she had to further analyze, and there she had done it with the HPLC. The same eluate had to be processed, then purified further with the HPLC. So there she got the 3 different picks at 3 different retention time point, and all those 3 she had to process to synthesize a nanoparticle and further use those nanoparticles for the biological activity, for the anti-cancer activity. So that was shown. Once you carried out the analysis, you could see that quite a fair number of various compounds were doped in the crude extract of the whole fruit extract, phonosopril, and the glycine, this raw glycine, alanine, and glutamine. Glutamine, glycine, and alanine were the tripeptides. This was very unusual tripeptides, which have been considered presently as therapeutics, whether it is antimicrobial as well as anti-cancer. So that was also coming along with this, and that as well as phonosopril have been documented to have a role to play in certain cancer. So that was interesting. Then she also prepared the another characterization of the particles produced from the HPLC fraction 31. There also she could get more of the di and a tripeptide of the oligopeptides. So it shows that oligopeptides seems to have a more role to play as an anti-cancer. So round-shaped mono-dispersing nanoparticles were synthesized using the whole fruit extract that played a reducing role as well as a doping agents on the surface of the nanoparticle. Anti-cancer activity seen both in the whole fruit extract as well as a whole fruit extract mediated green synthesized nanoparticle. However, the activity was more with the extract. Bioconjugation of green synthesized nanoparticle with arginine or aspartate cause a loss of anti-cancer activity. So probably the molecule, those tripeptide were masked by this, particularly converted into tripeptide or tetrapeptide, functionality of the earlier doped molecules. Then tripeptide as well as hexadecylamine were confirmed to be a phytochemicals doped onto the nanoparticle, especially from the HPLC purified fraction. The second plant is a Tridax procundum. Again, this is quite common in a semi-arid area, and especially in JNU, everywhere you see this plant, everywhere. It is commonly called as a coat button or a Tridax or a daisy also, and is a species of the flowering plant in the daisy family. It is the best known as a widespread weed and a pest plant. I personally have an interest in this plant because since my childhood, in Marathi it is called Dagadicha Fool, Dagadicha Fool or Dagadicha Bharas. It is, in Hindi they call it Jakhanjali. So it is having an application in healing the wounds. So that I experienced in my whole childhood. Every time whenever there was a cut or there was a wound, it was used very robust. Nurses had to crush the leaves and the bandage, you cover it up, and after 3-4 days, mostly it would be recovering. All over India, you can find it. All over India. I'm not so sure about Kashmir part, but semi-arid and dry area, you'd find it very easily. In JNU, probably on the ground, it's 90% available everywhere. Okay. So, we thought like probably on the skin part of wherever the epithelial cells are there, probably it has an effect. So it was also considered noxious because it spreads very fast, as well as therefore it is also considered as a pest. So common as Indian traditional medicine is anticoagulant, hair tonic, antifungal, insect repellent, bronchial cataract, diarrhea, dysentery, and wound healing. So I only knew wound healing, but after reading literature from the Indian text, I could figure it out. It also has been implicated for a systemic application. So there is no harm in trying something else on beyond the oil path or the skin path. So nanoparticles were synthesized, and those were in the range of 20 to 25 nanometer in the size. Again, monodispersion, spherical to oil-shaped nanoparticles were there. XRD shows again there is no contamination or there is no kind of a agglomeration of the nanoparticle. Zeta potential also shows that 20.7 millivolts, so that shows the stability of them. And we have been using the nanoparticle preparation for almost as old as a 3-year-old and having no loss into the activity. So FR also shows carbon-carbon single bond, NH, CE, OH, etc. All functional groups are essentially present in the extracts of it. Then the MTP was carried out with the A549. In the previous also it was used on the A549 lung carcinoma cell line. And here it is clearly seen that when we have the nanoparticles, then the middle one is the extract, and this is a chemically synthesized nanoparticle, and this is the silver nanoparticle. So at 42.32 microgram, it was having the IC50 value. So that's a quite impressive as compared to the previous experience that we have had with other plants. And vinblastine has been used as a control molecule. So here we could see that silver nanoparticle has very high IC50, that is 11,596 microgram, while the TNT, that is Tridex nanoparticles, IC50 is impressively small, that is a 42.7. Whereas the Tridex per se has a 40,676. The scenario is reversed than that of the Solanum xanthocarpum. Solanum xanthocarpum plant had activity in the fruit extract. Nanoparticles had reduced activity. Here, the nanoparticles have impressive activity. Plant is very bad as compared to the chemically synthesized. But even the chemically synthesized are 900 times weaker as compared to the green synthesized nanoparticle. Now, biofunctionalization was carried out again with the aspartate yellow color and the cyan color, the as arginine, and it is consistent that biofunctionalization does not necessarily work even in this case also. So whatever phytochemicals are there, if they get in contact with this arginine or aspartate, they may produce ionic or covalent bond and changes the structure of the functionality is no more retained. So here is IC50 again, 11,596 for silver nanoparticle, 41 plus for the 42.7 for the nanoparticle TNPs, 40,000 plus for the Tridex, then aspartate 6,000 and arginine 1,000, but still it's far lower than that of the nanoparticle process. So the agent is carried out for the further purification, as I told you in previous. The fraction 63 was processed further, and like in the previous case, the whole aqueous extract nanoparticle synthesis with HRMS was carried out to figure it out. So there were like 12 different compounds in the found onto the different type of nanoparticles, and Phonesoprene as well as tripeptides, already I have said that they have indicated to have a role to play in certain cancerous cases. So HPLC purified fraction was also utilized in the aspartate tyrosine dipeptide as well as acid modified with aspartate was also found. So interestingly, in both different cases, we have been able to find di or tripeptide either as amino acid as such or the modified amino acid at N terminus. So chronogenic assay was carried out using different concentrations, 2 microgram, 5 microgram and 10 microgram. It is evident that at 10 microgram there is no growth at all, and the concentration dependent, the effect is noted just 2 or 3 minutes. Okay, to tell the exclusion was also been carried out to the cell viability at different time point, 24 hours, 48 hours. And here is also another cell line, that is a mouse skin. It makes a very huge impact, and IC50 value is around 5 to 7, in between 5 to 7 micrometer, even more impressive than that of the lung particularly. Now, to figure it out at molecular level, what are the genes or what are the pathways or what are the processes being affected by the nanoparticle in the cancerous cells. So this is to show that the RNA sequencing was carried out, and there have been promisingly 32 genes, of which 14 genes are upregulated, while the 18 genes are severely downregulated. These are the highly stringent approach used to figure out the lesser number of genes. Otherwise, there are a couple of hundred genes which are upregulated and a couple of hundred genes which are downregulated. These are highly promising ones. And when we study the pathway, we can see that the KIT1 and KIT2 is also targeted, which has been known to have a role to play in anti-cancerous property. Then GPCR couple receptors, that is also one of the targets by this nanoparticle. Then the third is collagen. So that is also the ECM matter is also being affected in the cancerous cell. Then also the metallothionein, particularly zinc transport, and zinc has been known to play a significant role in the transcriptions, etc. So that is also a part of it. Here are the processes. There are more than 30 different processes being targeted by the nanoparticle. Here are the financial assistance from the DST and ACRB to be acknowledged. The cancer work initially started with the help of Professor Rana Pratap Singh's group, and Rani has completed her PhD. Now Nilesh is completing his PhD in the same topic, and Surbhi is a postdoc working in the same area. Thank you for your patience.
So I think in the interest of time, I would request the audience to hold their questions for the coffee break. Thank you so much, sir. And again, I would request Professor Anju to kindly felicitate. Thank you so much, sir. Our last speaker for the session is Dr. Vilas. Dr. Vilas is a senior scientist at Chittaranjan National Cancer Institute, and his work is on cancer genetics. Dr. Vilas has over 25 publications in international journals, and she has done incredible work on molecular targets of therapeutics for breast cancer and HPV.
Good evening. Myself, Dr. Vilas Nasare, Senior Scientist at Chittaranjan National Cancer Institute, Kolkata. So, differential expression of mitotic checkpoints protein and its clinical impact in advanced ovarian cancer patients. So, mitotic checkpoints protein means MAD1 BUB genes. That genes we want to check it. Is there any adverse impact or impact on the clinical sites or not, that we can check it in ovarian cancer patients. So, ovarian carcinoma in our hospital, we can say that a lot of patient will come from the persistent abdominal swelling, bloating, pelvic pain, vaginal bleeding, alert bowels, habits, embarrassment, loss of appetites, and weight loss. This kind of symptoms came, and then most of the patients, 2, 3, or 4 grade stages patients. So, for treatment therapy, we have given the carboplatin and paclitaxels. And intraperitoneal regime, we have given the paclitaxel and cisplatin as per the NCCN guideline 2020. So, our background is the spindle assembly checkpoint. If you have spindle assembly checkpoints, core protein is like MAD, BUB, and CDC20. This core protein is deregulated, it leads to the formation of tumorigenesis. So, our target is if this deregulated, so same pathway shared with the biological pathway activity by carboplatin and paclitaxels. So based on that protein, if they dysregulated, what effect on the thermogenesis, safety and prognosis of ovarian cancer, that we can check it. So this is the figure which we have already published in Chromosome Research. When the SAC is on, single assembly checkpoint, SAC is on, the CDC mitotic checkpoint molecules, it's a good relationship with this, and no anabase once it is there. So we have taken for this 76 patients we have taken for this study, and After the tumor size is large, then we will go for the neoadjuvant chemotherapy. After 3 cycles, tumor size is reduced, then we will go for the surgery. Yeah, if tumor is very small and it will easy to operate, so we will go for the directly surgery. And then we will collect the tissue for the ISC preparations. Then we will, this is the hematological marker like CA125 CT, radiological markers. Based on that, we have differentiated the patient responder as non-responders. So this is for this method, we have done this clinical endpoint efficacy and the side effects of the chemotherapy regimes. That clinical part, clinician did it, ISC, mRNA analysis and microRNA analysis, and then we see the bioinformatics part also, with these proteins. So if you see the presentations, this total sample patient is 76 patients, and as per FIGO guidelines, grade 3 and grade 4 is maximum 82 patients, 82% patients, and most of the patient is a serious pulmonary 81% patients, and gross type you can see that solid cystic patient is 58%. So this is the pre-treatment CA125, CEA and CA. These all are hematological marker. That marker you can identify the level of aggravating. It means it's a grade 3 or grade 4 markers, and then we will see the surgery. This is a interval debulking or primary debulking. This is a 54% we have taken. Means neoadjuvant cases and adjuvant cases. This is called as for surgery point of view, like the primary debulking and interval debulking. Then we will see the clinical response. What kind of response? Carboplatin and patination is given. So we have seen that clinical point of view, the complete response is 37%, for partial response is 40%, and objective response rate, complete response plus partial response, total response is 77. Other parts is the considered as a non-responder, like the palliative treatment to time to treatment and not available, like that. So now we compared with this adversity of the drug reactions. So we have seen that maximum cases we found in a grade 1 and grade 2 case isme, and the thrombocytopenia. This is totally out of 76. We have seen that anemia is a 65 cases. We have seen that leukopenia is 16, and granulocytopenia 27 patients. Constipation is 44 and indigestion 47%. We have seen that. So after that, we survival rate, we have checked it, the non-responder, responder and partial responder. So you can see the headline, you can see the step down the cases, most of the, so non-responder cases. It means the death rate happened most of the non-responder cases. It means the drug is not working well in these cases. So and the responder cases, you can see that only for 2 or 3 cases we died here, and other 6 partial responder cases it's high. And if you call as a long test, you can see that it's highly significant. So now this part is a completely clinical part. Then we will go for the immunohistochemical parts. That parts we can see that A and B, it is a part of the, this is the histological staining, and it confirms from pathology point of view, it's a ovarian carcinoma, and then we can see that C and D, it's a highly expression in the cell and nucleus and cytoplasm also. Then this is the MAD1 also expression in both nucleus and cytoplasm. And then this is the BUB1 gene. We can say that expression of nuclei is low, but cytoplasm expression is high. Then BUB3, this is the BUB1, BUB2, BUB3, MAD1, MAD2. It should be a little bit confused, but I will explain later also. So BUB3 expression pattern also, so cytoplasmic expression is there. So now we correlated this immunohistochemical expression with this responder, partial responder and non-responder. We have seen that there is no MAD1 and MAD2 as well BUB1 and BUB3. There is no correlation with each other. So then we will see the mRNA expression. mRNA expression, you can see that MAD1 mRNA and BUB1 above 36 above, you can say that the positive expression pattern is slow. So there is no as such correlation with it with each other. So next slide. Then we will correlate with this adverse interaction with this BUB1. We are MAT2 genes. Then we can see that the anxiety, abdominal pain, and constipation, it's highly significant with the BUB1 gene. And MAT1 was significantly associated with the weight loss and constipations. Then analysis revealed that BUB1 and MAT1 were 10 for -10 dominated than MAT2 and MAT3. So if you see the risk factor point of view, so you can see that BUB1 and BUB3 expression had higher risk to the survival than MAT1 and MAT2. Then we will see the bioinformatic point of view, okay, what can have happened. Because this is the patient and we can see that the genomics aspect. So this is a small part I just presented here, but now part is still pending. So now we will go for the bioinformatic stuff. And this is 2.5 years completed and half years most of the COVID में चला गया sir उसमें. So this is the MAT2 with the carboplatin. What kind of reaction? So you can see that this is the protein target, means MAT2, and then carboplatin. We can see that only 1 or 2 molecules bind with this target to the drugs, and then you can see that the paclitaxel also 1 or 2 protein amino acid bind with this drugs. Then you can see that after. If you see the inside the body, what kind of interaction will there, that we will check in the post-molecular docking stimulations. Based on that, you can say that most of the drug with the lot of proteins interact with each other, and based on that, you can see the edge bond pattern also find the range between the 2 to 7 in between. That means in 100 nanoseconds, you can see the number of interactions is high. So same thing in the paclitaxel also. Paclitaxel also interact with the after post modulations so you can see that the interaction is high. Then we can see that pH bonding how kitna hua, that we will check it. You can see that the aspirin and ASPA and ACR, a lot of H bond colors hai, that H bond, you can see that lot of interaction with the different amino acids. And same thing in paclitaxel, and this is the root mean square deviations. It means the interaction within a 900 seconds में कितना time हो सकता है, that we will check it, and you can see that carboplatin में अच्छी तरह से नहीं हो पाया. Okay, but in case of paclitaxel, you can see interaction is fast and it's very close. Yeah, you can see that. Then we finally table is concluded. The minus 7.331, the binding energy, if you see with the carboplatin with the MAT2L1 wild type, then you can see that this energy is so high, so higher score. So clinical observation, you can see that there was significant difference of clinical outcome, efficacy, safety and survival in ovarian cancer patient receiving carboplatin and paclitaxel. Molecular observation, you can say that, so molecular analysis, the SAC component have dysregulated expression in ovarian cancer pathology, the protein drug interactions lead to several specific adverse effects in patients. MAT2 expression may not be impacting chemotherapy response, but has a strong correlation with the poor overall survival. The SAC could be targeted for the identification of molecular difference between responder and non-responder to personalize the treatment and develop the novel molecules for higher grade serious ovarian carcinoma. And in bioinformatics point of view, in silico analysis reveals that stronger binding energy of MAT2 with carboplatin than paclitaxel. However, the paclitaxel is a more stable complex than carboplatin with the said proteins. So future plan is to identify genetics and epigenetics dysregulation in certain complex cancer pathophysiology and to develop the therapeutic approach by targeting SACs. This is my goal, And Sinjari Sarkar, this project is funded from DHR, and Sinjari Sarkar is one of the girls, so she is working on that. And thanks Dr. Santosh Kumar Behra and Sunil Kumar, this all the bioinformatics person did it. And this part is already just considered annals of oncology is sadly considered this part, and many other parts also included in that paper, but this is one of the part included in this paper.
Did you correlate survival with histological differentiation of children?
Sir, yeah.
You look at differentiation, like one were well differentiated.
Well differentiated because this is our hospital is a tertiary care hospital. So most of the advanced cases is coming. So grade 3 and grade 4 more be differentiated, and stage 3 and 4 cases generally आता है.
So survival poorly differentiated का अच्छा होता है या
अभी ऐसे है.
सबसे
सब चले जाते हैं.
Do we have any more questions? Thank you, sir, for your talk.
Thank you.
I would now like to request Professor Anisha Vastav to kindly felicitate our speaker. So I would now like to thank everyone, all the speakers and the audience.
I also have a pleasant duty to be.
Yes.
So I would thank all the speakers for such wonderful talk. It was intellectually stimulating. And I also thank impromptu chairpersons, Ms. Swati and Bhavika. Thank you, you've done a good job, wonderful job. Your plaques will be given later. Thank you.
So we invite everyone for tea outside. Thank you.
Good evening. I welcome you all to session 3 of the first day of the conference. And the session 3 is on emerging anti-cancer phytonutraceuticals, and it will consist of 1 keynote lecture and 4 invited lectures. I would like to welcome Professor S.A. Das on behalf of the whole organizing committee to chair this particular session. Professor Das is a senior consultant, Northeast Region Biotechnology Program. He pursued his PhD in tumor immunology from Banaras University and then went on to pursue his postdoctoral concept under the DAAD concept program from DKFZ, Germany. He has been a visiting scientist in the Institute of Molecular Biology and Biochemistry, Free University, Berlin. Sir held the post of professor in the Department of Biotechnology, All India Institute of Medical Sciences, New Delhi. He has also been a recipient of ISRO UNESCO Visiting Fellowship in the year 2021. I welcome you, sir, once again, and I would like to request you to take over the session. Thank you.
Can we request the audience, because I see only the speakers.
Yeah.
Okay, thank you, madam.
Start the session.
I welcome you all to the last session of the day. And first speaker of this session is Professor Damodar Saluja. Professor Saluja is PhD from Delhi University. She was assistant research scientist at New York University Medical Center. And she is joint director, Delhi School of Public Health, University of Delhi. And she is officiating director of B.R. Ambedkar Center for Biomedical Research, University of Delhi. Dr. Saluja, please.
This is like a no-show movie
No, ma'am.
where there are no audience. So everybody either is too tired since morning or they are enjoying their tea. So
You can wait for me.
yeah, they must announce in the tea session that now the sessions have begun and Dr. Neeta Sagar is also not there. She has not.
She has not come yet, yes.
Thank you. All right. This is what happens normally afternoon sessions. Everybody starts feeling tired and exhausted. But never mind. So we had been looking at one of the natural compounds, ascolitic, for quite some time now. But not very enthusiastically in the beginning, only very recently we have started working on it a little more. So ascolitic is a coumarin compound, and as you can see, a lot of plants have this compound, and in many of the Himalayan region areas or high altitude region, many people were using these plant decoctions for several diseases. So when we looked at the literature, we definitely found antioxidant activity of this this compound. So you can see that many antioxidant activities were reported as early as 1883, 87, 90 and 95 and all that. And later on, people also observed that it has it has cytotoxic activity and inhibits apoptosis, and kind of has some, it affects apoptosis and many other activities. And our interest was more in the leukemic cells, because we had a model system in lithium translocation in Cosme cell lines, which have 821 translocation. And that is something very interesting, translocation. And in fact, in many of the leukemic patients, that comes back, they can have any type of leukemia, but residual disease comes back with this 821 translocation. So that makes this translocation somehow important in the clinical setups. So we also found that some of the other cases, as we started looking into 2002, 2003, 2006, you can see that people had started reporting its effect, anti-proliferative activity, basically into various cell lines. And then in our own lab, we looked into the Cosme 1 cell line. And if you really look into this global burden, so global burden in 2017, it was observed that there is a gradual, in the last 3 decades, there is a gradual increase in leukemia. Why, that is not very clear, but definitely there is a global increase. And if I show you this graph, so you can see India, China, and North America, they seems to have a higher burden of leukemia in their, although many of these are, North America is a high income group, but still there was death associated, there were, incidence was higher, and there were many such manifestations, as I was telling you, that after in any kind of leukemia, MDR becomes a minimum residual, so MRD becomes a common feature. So as you all are biology students, you know that, all of you know that leukemia is basically loss of differentiation of the hematopoietic stem cells. And of course, it could be acute myeloid leukemia or chronic myeloid leukemia, or it could be acute lymphoid leukemia or chronic lymphoid leukemia. And basically what happens is that there is a proliferation of the cells into the undifferentiated cells will proliferate and result in formation of the blast cells, mainly myoblasts. And this results in the malfunctioning of all the activities of the blood, and gradually the anemia and cytopenia and neutropenia and all of other diseases, all other symptoms will set in. So we also all understand that leukemia or any cancer for that matter is not one, normally not one hit hypothesis. So this is well established. So the primary gene mutation could be translocation, but then there are secondary mutations, and some of the common secondary mutations that have been seen is in the FLT3 and in c-KIT gene, which are both are into the differentiation proteins, which tyrosine kinase activity and other kinase activity. So we know that these are there. So I can also say that there are 2 sets of genes which are important in any cancer, including leukemia. There is loss of function of transcription factors which are required for differentiation, and gain of function of signal transduction molecules which are responsible for higher proliferation. So both together leads to leukemia or any cancer that we look into. Okay, and AML ETO is not any different. So if we look into the current therapy of leukemia, We do not have any specific drug for acute myeloid leukemia. Although several papers have started coming up in the last 1 decade or little more than that, where natural compounds have been shown to have some effect on the chimeric gene expression, which is very important. Like that means AML-ETO translocated mRNA is affected. Whatever else is affected is, of course, is a matter of study, but AML-ETO translocated gene expression was affected, and that was a very important contribution. Okay, so and of course some others will result in caspase mediated apoptosis, whereas oridonin, which is another natural compound, and which results in the AML-ETO and c-KIT expression analysis. Why I am emphasizing on that is that taking lead from these compounds, we also started looking into escolitin and how that affects. And we also observed that this coumarin is responsible for both anti-proliferative activity on COSME1 cells with 821 translocation, AML-ETO. So AML is a very important transcription factor for differentiation. And ETO is a suppressor protein. So when you have the mechanism as it is considered is or conceived is that AML-ETO chimeric protein, instead of activating transcription, it represses transcription in a specific manner because DNA binding domain of AML is retained. So all AML genes will be downregulated because of this chimeric protein. So this will have important consequences when I talk later. So as you can see, escalating induces growth inhibition and cytotoxicity in the COSME1 cells, and cell cycle arrest in G1S phase that we observed. And this was further, checked by caspase-3 expression and all the caspase-8 and PLC gamma expression increases. And so we did all the classical experiments to prove that it is a change in the apoptosis and cell cycle and everything. So as you can see here in apoptosis, both early and late apoptosis was observed in the cells. So you can see here the late apoptosis and early apoptosis was observed. So having established that escalatin could be a good anti-proliferating compound, I would not say anti-cancer drug because it will be pre-assumption. So anti-proliferating compound, we went ahead to look into the expression of AML-ETO because I remember I told you oridonin has this quality of decreasing the levels of AML-ETO chimeric gene, but AML levels were not affected. How we do not know, and here also we observe both at the RNA level and the protein level, which I will show you in the next slide. In fact, what we observed was they were decreasing, escalatin was decreasing the half-life of the chimeric mRNA, whereas it was not affecting the half-life of the AML normal transcription or mRNA was not affected. Not only that, even the c-KIT expression was high in AML-ETO, but that also when we give the cells escalatin, the half-life of the c-KIT was also decreased, and within 2 to 2.5 hours, you almost see negligible expression of both the chimeric mRNA and the c-KIT expression. And this was further supported by at the protein level. As you can see that AML-ETO and we did not see c-KIT expression using specific antibodies. But further, if that is happening, then the genes which are regulated by AML should be regulated, right? The normal AML. That was I told you that AML-ETO act as a transcription repressor rather than transcription activator. And we did observe that. So LAT1 and RUNX3, which are regulated by AML normal AML gene or transcription factor to C, they were upregulated when we gave escalating treatment. So you can see here good expression of this, and this is vehicle control and this control. So we had a gap after this study, and we had due to various reasons, and then very recently, so that part was, most of that was done by Dr. Sharad Sani, who is in Thermo recent now. And then Dr. Ankit Mathur joined my lab as a postdoc, and he had the interest to look into this problem further. So what we decided to look into the effect of escalating on the differentiation. Now we are seeing that AML ETO gene is inhibited. CTD is also inhibited. Does it reverse? That means, can we achieve the differentiation of hematopoietic cells back to, that is what is, I told you, the leukemia is undifferentiated myoblast cells. So he started looking into that. And of course, there are leukemic stem cells and within the blast cells, and that's another story that we are looking into. But I will not talk about that here. So we started looking into differentiation therapy, whether it causes differentiation. Now, one of the reasons for that was that very in the last couple of years, all trans-retinoic acid was being used for this kind of a therapy. So when he started looking into this, to our surprise, we found that escalating does promote differentiation in the progenitor cells, as you can see here. So there is increased cellular granularity, there is reduced immature marker, which is CD34, and there is enhanced maturation marker, CD38. You can see here the gradual decrease in the immature marker and gradual increase in the maturation markers with the increasing concentrations. So we then also went into the expression of other CD11B, CD18, CD44, CD33, and we did find that in all these cases, we did find that there was increased differentiation, and the differentiation was targeted more towards neutrophils. But we did not see much difference in the monocyte lineage markers, as you can see here, plus minus escalated. But neutrophil markers were altered, and as as shown in my next slide, we also saw in the blast cells, there were all the features which are normally present in the neutrophil cells, like, lower nuclear to cytosolic ratio, and there was a bilobe structure and increased vacuole formation, vectorization, and also, azophilic granules were there. So this also supported our hypothesis and with the marker that there was a differentiation of these cells into neutrophils when we give escalating to these cells. Now, we also know that we also understand in many of these differentiation and proliferation, Wnt signaling plays a very important role. So we also looked into Wnt signaling to understand the mechanism. So if you look into general Wnt signaling, everybody knows there is a canonical pathway, there is a non-canonical pathway. Canonical pathway is normally characterized by beta-catenin levels, and non-canonical pathway we normally talk about calcium-mediated regulation of the non-canonical pathway, and it results in the planar cellular polarity. Okay, so we looked into taking the RT2 profile assay. We looked into almost 84 Wnt signaling genes using that plate assay, and we found that we had a number of non-canonical pathway genes which were getting upregulated. So we went ahead and carried out straightway real-time PCR just to reinforce the important expression of the important genes. So we, as you can say, DKK1, which actually inhibit canonical pathway but activates non-canonical pathway. And there are other proteins which are involved in many of these non-canonical pathway, and also the genes which are involved in the self-renewal, they were almost inhibited in the presence of ascolitin. Okay, so this encouraged us to look into more into the, so obviously as I told you, calcium plays an important role. So we started looking into the both beta-catenin expression and the effect of calcium. So first we checked the expression of beta-catenin in the presence and absence of ascolitin. As you can see, there was decrease in the beta-catenin expression in the presence of ascolitin. And then when we looked into the calcium levels, they were definitely increased by the ascolitin. Now calcium in the cell could increase, these are the other genes which we looked into, which are important in the differentiation, which are downstream of the Wnt signaling pathway. So as you know, calcium can be increased either by the influx through the transmembrane or due to ER. So we wanted to differentiate between the two mechanisms, and there are specific inhibitors which are known to cause inhibit one or the other pathway, and so this is actually we blocked the transmembrane calcium influx using amlodipine inhibitor, and we found that although calcium pool decreased, but when we added the ascolitin, the calcium pool increased again, and we did not see any difference in the effect, whatever ascolitin effect we were seeing. So as you can see here, ascolitin and the inhibitor were as good as ascolitin itself. Okay, and same was true for if we were to use the this marker CD11B, the differentiation marker. Okay, so so this was clear that the transmembrane calcium was not very important. So then we went ahead and used the inhibitor for ER calcium using TMB8, and we found that now we do see the loss of effect of ascolitin in the presence of the inhibitor. Although there was slight increase upon the inhibitor alone, but this is almost equivalent to the control, as you can see, right? It could not come up to the ascolitin. This increase was not observed. Similarly, when we looked into the CD11 expression, 11B expression, here also it was more like control, and it could not overcome basically. So we knew that now this ascolitin was targeting the calcium levels through ER pathway. So, so just to summarize up to this point, so there was a increase in CD11B, increase in CD38, CD34 was decreased, so there was increased differentiation when ascolitin is there. But when amlodipine was used, it did not affect. It was more or less like control, ascolitin alone. But when TMB8 was given, then we could not see the effect of ascolitin. So basically, ascolitin mediated differentiation was inhibited. Now, a very important point that is there is DKK1. And we know that TGF-beta has a very important role to play in regulating DKK1. Now, DKK1 or TGF-beta mediated regulation of DKK1. So TGF-beta will result in the upregulation of canonical pathway, but inhibiting the non-canonical pathway, right? So what will happen is, So if we do that using TGF beta, then we should not be able to see the effect of escalating because TGF beta is inhibiting, so we will not, and that's what happened. Escalating alone could give us upregulation of the differentiation marker, but when we use TGF beta, it is inhibited, and TGF beta and escalating could not bring about the escalating effect. That means TGF beta was inhibiting, escalating mediated Venn axis was inhibited. Okay, so this further proved that we are looking into Venn signaling axis, and non-canonical pathway was the most important one. So we also, just to be even 100% sure, now we inhibited the canonical pathway. Right now, using TGF beta, we had inhibited non-canonical. Now we inhibited canonical pathway to see do we get better effect of escalated. And sure enough, we did see a better effect. So if this is escalated, we add the inhibitor. So inhibitor alone is more like control, but inhibitor plus escalating result in a better effect of differentiation marker upregulation. And also more of inhibition of the beta-catenin degradation is there. So based on all these studies, what we propose is that the escalating moderate DKK1 for sure, and it somehow get translated to calcium levels of ER-based calcium levels, and this results in the upregulation of the differentiation markers and downregulation of the proliferative markers by modulating beta-catenin or degrading beta-catenin. Although the intermediates may not be very clear at this point, but we do understand by showing up from our results that beta-catenin looks to be degraded, and of course there is a very clear-cut pathway. In addition to that, we had, as I told you, that we had also observed that escalating is responsible for degradation of the chimeric mRNA and result in the differentiation of the leukemic blast cells to the neutrophils. Okay, so that means escalating, we are not saying that escalating can be used as a drug itself, but it could be as an adjuvant, and more research, of course, will be required. A lot of animal preclinical and clinical studies will be required to see whether it works actually in vivo conditions, because what we have done is with the cell lines. And therefore, it is very important that we look into some of these natural compounds, which can not only result in cytotoxicity, but can also target the primary mutation, the expression of the AML-ETO, but also results in the change in the pathways which are responsible for proliferation, right? Your Venn signaling pathway. So, there is some, we had also done some work on the antioxidant activity, which I am not describing here. So all this work was done by Dr. Sharad Sani in the beginning, and more recent work was done by Dr. Ankit Mathur. Thank you for your attention, and we received the funds from DST and DBT for this. Thank you. Any questions? I will be happy to answer.
Question, please. Yes.
एक्सप्लेटिंग जो होता है वो यूनिक क्लास को प्रोफेसर में ट्रांसफर करता है.
Yes, we don't know whether it is transferring the leukemic stem cells into neutrophils or it is differentiating the cells, because we did observe even the stem cells getting differentiated. But those are very difficult experiments because you require cell sorter and removing those, culturing those cells. So we have not been able to really do too much study on that. But it looks like that in the cell culture also you see a lot of leukemic stem cells. So, so if you use certain growth media, you can enrich those cells. So we think it might be responsible because one thing is good in the blood because they have a restricted life. So we think that if we don't worry about whether myoblasts which are pre-existing, they get differentiated or not, but if the leukemic stem cells will get differentiated, then we will not have further proliferation, right? But those experiments we need to do it in a very specific manner by culturing only the stem cells and then looking into these effects. Yes. We don't know. It's a difficult question, very good point, but it is, there's nothing in our biology which says why. This is what we observe. So also remember these cell lines are growing in the laboratories for last so many years, right? So we don't know whether it is just that there are inherent mutations that it cannot differentiate into other cells. Maybe if we are able to get some primary cell lines and stem cells, primary stem cells, leukemic stem cells. We are submitting a project for getting samples from the patient and then looking, but the problem comes is you need a naive sample who has not been treated with any drug. So from there, if we isolate the stem cells, so then we can answer probably your question. We also might be lucky to differentiate into various subtypes. That will be really good. Yes.
So, if there is no question, thank you very much, ma'am.
Thank you
for your nice talk.
Thank you for your attention.
Kindly request to felicitate Professor for this enlightening talk.
Next talk is by Professor Prabodh Das S. Patel. Dr. Patel is in Cancer Biology Department of Gujarat Cancer Research Institute in Ahmedabad. His interest is in the area of role of glycosylation in relation to development and progression of different types of cancer, mainly tobacco-related cancer, BCR-level independent molecular mechanism of imatinib-resistant CML, and also salivary glycosylation to establish saliva as a non-invasive diagnostic tool for oral cancer. Professor Patel has done PhD from Gujarat University. He has been visiting scientist at Nebraska, USA, visiting scientist at UCLA, California. He has about 150 original publications and he received numerous awards, prestigious awards and honors. Professor Patel, please.
Thank you, Professor S.N. Das, for the introduction, and good evening, all distinguished delegates. I take this opportunity to thank Dr. Alok Bharti and Professor Anju Srivastava and the team for inviting me to this well-organized conference. And I'm going to talk about our work on oral cancer. In next 15 to 17 minutes, I am going to tell you and convince you that oral cancer is a major health hazard caused due to tobacco consumption, and it has very well defined molecular progression pathways. Our research on glycosylation and other hallmarks says that it needs multi-targeted approach, and nutraceuticals or natural polyphenols are the answer to it. And there is a lot of published data on this topic. So oral cancers are arising in oral cavity involving all various sites in the oral cavity. Most of these cancers are associated with high rate of tobacco use in our country. It ranks third in the cancers with high mortality rate, with an estimated death of around 76,000 per year, as reported by ICMR. It may not be important globally because it is 9th highest incidence of oral cancer in the world, but it is important for us to address this issue because it is third most common cancer in India. Moreover, even in the primary or a localized cancer, survival rate, 5-year survival rate is around 55%. The problems associated with this disease are late representation, local regional spread of the disease, recurrence or fast recurrence, low overall survival, no targeted therapies, and very alarming situation is that the incidence trend of the disease are shifting to the younger population. As I said, it is a tobacco disease or a tobacco caused disease, and as reported by ICMR, these are the states where oral cavity cancers are high, and Gujarat is one of them. And as per global audit tobacco survey, around 30% of the population has tobacco consumption in various use. For oral cancer, non or a smokeless tobacco is the cause of the disease. So question is how to address this issue. There is exposure of tobacco carcinogens, and there is a gap between onset of the disease. One can have markers or device for screening of the population which are at high risk. For secondary prevention, before onset of the disease and after onset of the disease and onset of the symptom, secondary approaches by early detection can be helpful. Ideal biomarkers when the disease is progressing or in an early stage for monitoring disease progression can help. And if there are multi-targeted therapies or good targeted therapies, recurrence can be stopped or delayed, and the disease may be cured. So this is an ideal situation. But let's see whether it is successful. Yes, it is successful in case of cervical cancer and other cancers. There are markers for early detection, pap test and mammography. For tertiary prevention, there are several biomarkers for various cancers, and there are various targeted therapies, and therapies like imatinib in leukemia has changed the scenario. But anyway, you will not see oral cancer. So what is the scenario in oral cancer? For primary prevention, we have only visual inspection. There is no marker for early detection. Wait till the disease manifestation. No ideal biomarker. There is only conventional therapy available, that is cut, chemo, or burn the disease, that is chemo, radio, and surgery. And there are no target therapies for oral cancer. And it is time to shift our focus to oral cancer, because as reported by Globocan, India has highest number of deaths due to oral cancer. So what can be done? For oral cancer, salivary diagnostic for early detection or for screening can be helpful. Biomarkers for high-risk group identification, better prognosticators, comprehensive drug targets, and ideal targeted therapies can be helpful to change the scenario. And if you have to do that, you will have to understand biology at each step. And if you look at reports on biology, there is very well defined or very well characterized molecular model from normal to mild dysplasia due to tobacco carcinogens, to moderate dysplasia to severe dysplasia, and carcinoma in situ and metastatic cancer. And at each step, there are very well defined or very well reported molecular biology changes. But to have a comprehensive view on biology of cancer, what one will have to do is to look at all the hallmarks or various hallmarks of the cancer, and that is what has been the focus of research of many researchers in recent years. But with the progress of science, hallmarks of cancer, still there is a road ahead. There are several unresolved questions, and one of them is significance of epigenetic alteration or PTMs, post-translational modifications, which may be looked or which may be addressed. And the major or the most prevalent PTM is glycosylation. And among neoplastic or neoplasia-associated epigenetic alterations, changes in cellular glycosylation have been recently received attention as they play a key role in disease progression. These PTMs are important because it is a small change at the genomic level, and due to post-translational and post-translation modification, it has a huge impact. PTMs of proteins influence enzyme activity, protein turnover and localization, protein-protein interaction, and so on. And the common modifications include glycoprotein ubiquitination, and so on. It is also reported that glycosylation, rather than a hallmark, it is looked into another perspective, that it play a role in every recognized cancer hallmark, and it is not only a hallmark of cancer, but it is an enabling characteristic of cancer. So we have a long-standing research work on glycosylation, and we also have published that glycosylation is another hallmark of cancer, and it is authenticated by Glycobiology Society. Recently, in addition to our earlier studies, recently we carried out a study on oral cancer patient, and these are the characteristics. The major sites of the cancer were buccal mucosa and tongue. I will not go into the details, but RNA extraction from malignant and adjacent tissues were carried out. cDNA, it was converted to cDNA, and RT-PCR or real-time PCR assays were carried out. In the interest of time, I will directly summarize the result. In our cohort, majority of the cancer patients were male, as it is a male-dominated disease, and among the tobacco-habituated patients, majority of the patients were exclusively tobacco chewers. So tobacco chewing is the major risk factor. The oral cancer patients, the mean age was 45, and as I said, it is shifting to the younger trends. It was good to note that tongue carcinoma was a primary, all the patients in the present cohort were squamous cell carcinoma with moderate differentiation and early stage of the disease. And out of the cohort, majority of patients had tongue carcinoma as a primary site of the disease. Our real-time PCR data on glycosylation changes were interesting. These are several cell-lyl transferases, that is cell-lyl transferases and glucosyl transferases Among them, ST3-GAL1, ST3-GAL2, NEU3, FUT5, FUT6, FUT-A1, MMP9, VEGF, BEX, and P53. They were higher in tobacco-habituated patient as compared to non-habituated patient. So tobacco and its metabolite are, so we interpreted that tobacco and its metabolites are the culprits leading to the altered glycosylation in oral carcinoma. Secondly, ST3-GAL2, FUT5, FUT6, VEGF-A, VEGF-C, and BCL2 were increased, and increase was associated with disease progression and lymph node metastasis. So we interpreted that they can serve as valuable indicator of the aggressiveness and metastatic potential of the disease. Thirdly, these were the transferases ST3, FT4, NEU3, VEGF, all those biomarkers or molecular signatures, they were upregulated in tongue carcinoma as compared to buccal mucosa, which suggests a role behind aggressiveness of the disease. So we try to correlate our data with our earlier publication as well as its role, its association with glycosylation. And what we concluded that increased glycosylation causes increased expression of MMPs, which is responsible for ECAD protein, loss of ECAD protein, which is further involved in increase in CJUN protein, and increased PEGFR is involved in upregulation of MMPs. So this is what we interpreted from our data on human cohort. And glycosylation was observed to be correlated with all other hallmarks. So we thought there are hallmark signatures and altered glycosylation. So stop wasting time around and just make a bound. We have our own gold mine of natural polyphenols. You know, everybody knows that cellular pathway which are affected by activities of nutraceuticals in various diseases. For precancerous lesions, they have anti-inflammatory modulators, and tumor spread, they inhibit tumor invasion, anti-metastasis, and so on. So they have chemopreventive as well as chemotherapeutic properties. No need to introduce what are natural polyphenols to this audience, but yes, they have effect on carcinogenesis process by promoting DNA repair, eliminating transform cells in disease promotion. They inhibit cell proliferation, anti-inflammatory properties, anti-hormonal effects, and for disease progression, they inhibit cell proliferation, inhibit angiogenesis, and induce apoptosis. So because of these properties, polyphenols have been useful in cancer prevention, chemo prevention, and as well as for chemo enhancer or radio enhancer. And these natural polyphenols, they have been playing role at all hallmarks of cancer, I would say. And there are many clinical trials. Unfortunately, there are very few or no oral trials on oral cancer. There are few oral trials on oral cancer in curcumin. So we collaborated with IIT Guwahati, Dr. Ajay Kalmukra, and we have studied the expression of various glycosylation transcripts in SAS cell line, which is a tongue carcinoma cell line. Experiments were carried out, and we have studied effects of various compounds in this cell line. We studied tobacco carcinogens for NNQ, NNA, and benzopyrin, because oral cancer is a tobacco-induced disease. We studied the effects of curcumin, picethanol, and butene as natural compounds. And we tried to correlate this data with the study, effects of cisplatin treatment. The cells were treated and incubated, and RNA was extracted, which was further converted into cDNA, and real-time PCR was carried out for the same parameter which we studied for human cancer. This is cell proliferative assay. I am taking you to the results directly, and this is a treatment to the cell line with tobacco compounds. This is 4-NNQ, 4-NQO, NNK, and benzoyapyridine in different concentration. And what we found was there was a dose-dependent increase in several transferases, MMPs, VEGF, with all the tobacco carcinogens. And what we observed in the patient was increased expression of all these molecular signatures. So it says that what was seen upregulated in tumors was also upregulated by tobacco carcinogens. Secondly, this is treatment with natural polyphenols, curcumin, picethanol, and butene. And here what we observed was curcumin and butene downregulated or FUT8, MMP2, and upregulated FUT3. These are the results of picethanol, decreased levels of ST3-GAL2, FUT5, FUT8, MMP, and VEGF. And if you remember, this ST3-GAL2, ST3-GAL3, FUT5, FUT4, FUT8, VEGF, they were worst prognosticator in our human data, and FUT3 was a good prognosticator in human data. So the data between cell line results and human studies correlates with each other. This is treatment with cisplatin, the chemotherapeutic compound. Again, this there was downregulation of dose-dependent effect of ST-GAL3, ST-GAL2, FUT5, FUT8, MMP2, and BCL2, and there was increase of FUT3 and BEX. In our human samples, these parameters, ST-GAL3, ST-GAL2, FUT4, FUT5, they were found to be worst prognosticator, and FUT3 was the good prognosticator. So here also there is a mirror image of the data between cell line studies and human studies. In brief, if I have to summarize this study, there was similar picture of altered hallmark signature between tobacco habituated patient and tongue carcinoma cell line treated with tobacco compound. The worst prognostic markers found in tissue study gets inversed in the tongue carcinoma cell line treated with natural as well as chemotherapeutic compounds. Targeting altered hallmark transcript in oral carcinoma, that is tongue carcinoma, with treatment of several natural compound alone or in combination with chemotherapeutic agents can aid in better management of the prognosis, and the data was very well taken by various journals. This is one of the publications. So we tried to put
all our previous and these studies together, and what we found is that there is no doubt that tobacco is a major risk factor. Oral cancer is due to late representation. Management is challenging due to late representation, and there is a high mortality which needs for better diagnostic and prognostic biosignatures. Aimed at molecular signature acts in synergy with the altered glycosylation transcripts of hallmark. There are several transferases like cell transferases and fucosyl transferases and MMPs as well as VEGFs as early molecular signatures of the disease metastasis and recurrence in tongue carcinoma. Altered transcripts of glycosylation, that is tobacco habit and disease advancement and site of the disease, is found in the patients. And the data on curcumin, butene and pacethanol says that they have high potential natural remedies against altered molecular events in the oral cancer. Also, circulatory markers of glycosylation and hallmark signatures can be used as liquid biopsy approach using saliva and plasma levels. So, we found these biomarkers as a predictive panel of tissue biomarkers. They are STGAL2, STGAL3, FUT3, FUT4, FUT5, MMPs and VEGF. The predictive panel of the circulatory biomarkers, so there is a tissue biomarkers and circulatory biomarkers because samples are not available after surgery in human studies. So, I mean, tumor samples. You may get blood samples or saliva samples. So, the predictive panel of circulatory biomarkers was STGAL2, STGAL3, FUT5, MMPs, and VEGF, and natural products to strengthen the armamentarium are pacethanol, curcumin, and butene. This is what we found, and we tried to look at the literature, and there is no need to say that curcumin is a very well reported compound, and this is a recent paper saying bibliometric analysis of the 100 most cited articles on curcumin, and they are all the studies promoting curcumin as a chemo enhancer or chemoprotector. There are reports on curcumin in head and neck carcinoma, anti-inflammatory effects, enhanced anti-inflammatory effects of resveratrol, effects of curcumin, and on NF-kappa B signaling pathways. There are studies on curcumin and tumor microenvironment in head and neck cell cancer, head and neck cancers. There are studies on butene saying that it has anti-inflammatory effects. There are studies on pacethanol saying that it is a chemokine inducer. Anti-inflammatory effects of resveratrol and other compounds. So there are so many studies to support our observation that these three compounds are can be added in the armamentarium. Molecular mechanisms for butene reflecting their chemopreventive potential. This is again potential of butene. And there is a whole lot of data on these compounds. So, to summarize, I think I have been able to convince you that there is urgent need to strengthen the fight against oral cancer. There are enough evidences to say ill effects of the tobacco. So evidences are enough now. There is oral cancer is progressed through various stages. So it is an ideal molecular marker to study carcinogenesis. If you ask me a question why natural polyphenols, so I'll say that stop wasting time around, just make a bond. We have widely published data on glycosylation, and study of biology is there at each step. Curcumin, butene, and pycethanol are promising natural remedies. So it is time to make best use of our own gold mine. Before I stop, I'll say that we had enough patients when we only had chemo, radio, and surgery. But now we have everything. So we have to have an attitude towards fight against cancer to make something different. Thank you very much for your kind attention.
The talk is open for discussion. Any question? Mr. Patel, have you done any xenographic study?
No, we have not.
Okay, thank you very much.
Thank you.
And thank you for keeping time.
Yeah, thank you.
I would now kindly request Professor Das to felicitate Professor Patel for delivering such an outstanding lecture.
I invite Professor Navneet Agnihotri for her talk. Professor Agnihotri is Professor and Head, Department of Biochemistry, Punjab University, Chandigarh. Her area of research is nutrition and cancer biology, lipid metabolic reprogramming and cancer biology. She is a doctorate from PGI Chandigarh, a famous institute, and she had been trained at MD Anderson Cancer Center, Texas. She is the recipient of various awards. Professor Agnihotri, please.
Good evening, everyone. Good evening, Professor Das and Professor Agarwal. And at the outset, I would like to thank the organizing committee to give me the opportunity to present my work and also arranging a fantastic conference. So the topic of my talk today is slightly different from what you have been hearing. It is about synthesis and evaluation of glutamine conjugated organotin-4 shift-based compounds as potential anti-cancer agents. So to this august audience, I don't have to talk about colorectal cancer, but colorectal cancer we know is the third most common cancer and the second leading cause of mortality globally. Though India has less incidence of CRC, but with the change in the lifestyle and the nutritional habits, CRC is coming up fast or emerging as a coming up cancer. Now, CRC, we know, comes from the adenoma carcinoma sequence, is very well established, and it occurs by mutation in various genes in a sequential manner, starting with APC, then you have KRAS and P53, and so on. As for the rest of the cancers, the treatment strategies here also include surgery, radiotherapy, or chemotherapy. And in the chemotherapy, what has been utilized is 5-fluorouracil or other pyrimidine analogs. This is a capacity bean, which is again 5-fluorouracil only, irinotecan, oxaliplatin, and so other drugs. But we all know that there are lots of side effects of chemotherapy, which includes, nausea, vomiting, fatigue, hair loss, bone marrow suppression, liver functions are altered, kidney function is gone, and so on. Now, with the advent of cisplatin, there was a lot of emphasis on metallotherapeutics. Now, metallotherapeutics is that you have the metal drugs which can go into the cancer and then has an anti-cancer effect. So cisplatin, is a part of the chemotherapeutics, and it's been used for almost every kind of cancer. And the kind of mechanism of action of cisplatin is that it acts at various levels and has been found to be very, very successful. But again, then there is a toxicity which is associated with this also. Now, what we started working on was with my collaborator in chemistry department, Dr. Virender Kaur, was on the organotin 4 compounds. Now, this organotin 4 compounds, the structural framework is very interesting because you have tin in the center, and on the one side you have the organotin moiety or the hydrocarbon groups which you can attach, and you can keep on changing this change. And then on the other hand, you can have a donor ligand which will help it to target particularly to the tumor cell. And we know that most of the side effects of chemotherapy are actually associated with its off-target effects. And organotin compounds earlier also have been people have worked on, and they found that it acts by various mechanisms which include mitochondrial respiration, alters the membrane permeabilization, causes apoptosis, also involved in the calcium-mediated apoptosis. So what we decided to do was that we decided to work on these organotin 4 compounds. And so again, we know that tin compounds also have a toxicity. So we started looking at it, that how can we improve their therapeutic profile? Can we understanding the inherent nature of a tumor cell, can we modify it so that there is less off-target effect and can be used as a better chemotherapeutic? So what we started doing was that we started looking at the metabolic programming in the cancer cell. My lab has been working on metabolic reprogramming, and we also looked at the others. And we know that glucose is one thing which has been exploited clinically also for PET scan and so on because of the enhanced glucose uptake by the tumor cell, and it's a highly sensitive diagnostic technique for the cancer. So what we did was that we started looking that beyond sugar, what does the tumor cell need? Then we come at this molecule, which is the glutamine. Now, glutamine addiction is again a very well-established fact of a tumor cell. And if you look at it, when a cell goes from low cancer goes from low invasion to the high invasive cell type, it switches from glucose to glutamine and gets addicted to glutamine. And why this glutamine is required? Because this works as a precursor for many macromolecules, which includes lipids, nucleotides. We know that it transfers the amino group for the purine and pyrimidine synthesis, also for the non-essential amino acids, then also hexosamine, then there is glutathione, and all of these, they actually require glutamine. So what we did was that we took glutamine conjugated organotin 4 compounds, and we tried to see that if we design an organotin moiety which has on it attached glutamine residue, can be targeted to the cancer, and can we have a targeted therapy and then can have this anti-cancer activity. And if we increase the targeting of the drug to the tumor, we can of course decrease the side effects. There will be tumor selectivity, there will be enhanced cellular uptake because of the glutamine residues, and of course minimal systemic toxicity. So this was our work done. So what we did was that we did the synthesis and characterization of glutamine conjugated organotin 4 compounds, and then we characterized by different techniques. Then we did the in silico ADME evaluation and molecule prioritization using the Swiss ADME method. We did the DNA interaction studies, protein interaction studies, and then we carried on to the in vitro studies for the colon cancer cell line. So this was the scheme for the synthesis of glutamine conjugated diorganotin for shift-based compounds. So this is glutamine, magenta, and then we kept on changing the side chains for this, for the donors. We had methyl, we had a butyl group, we had a naphthaldehyde group, then we had a benzaldehyde group, and so on. So we generated a list of compounds with exploiting this. And then we characterized whether what we have synthesized is the right compound or not, and that was done by FTIR, by NMR, by mass spectrometry, single crystal XRD, again tin NMR, 1H, this hydrogen NMR, and so on. And I'm not going to go into details of this because this has been already published in the Journal of Applied Organometallic Chemistry, and it is widely available data now. So from here, then we moved on to, so once we established a list of compounds, we made a number of compounds, and then we looked at it that which have the potential to be used as a drug. For that, we moved on to the Swiss ADME analysis. Now, in the Swiss ADME analysis, ADME stands for absorption, distribution, M is metabolism, and then finally is the excretion. Now, I hope this was clear, but what happens is that this is the pink box is the one. So any compound which can, this pink box basically represents the 6 physicochemical properties of the compounds. So if a compound fits within this pink box, that means it is bioavailable. So this is what it is, and we looked at this is the bioavailability radar graph of the drug likeness parameters. And what we see is that these are the different compounds which are generated 1, 2, 3, 4, 5, and then we took the 2 known chemotherapeutic drugs, which is the 5-fluorouracil and also the cisplatin. Now, all of our compounds except the 5 was slightly outside, but the rest were very easily bioavailable. Then another thing which we did was with the boiled egg method, though the white side is not there, but this is the yolk side, and the outside you have a white egg sac. So any compound which is within the white portion of the egg represents that it is not taken up by the brain. So the yellow portion represents that it is taken up by the central nervous system, and of course will be highly toxic because of this. So what we did was that there was none of these compounds, whatever we took, and also the cisplatin and 5-FU, there was no CNS uptake. In fact, and I think there was I hope the blue dots are clear here. These are the blue dots, and this is the red dots. And these blue dots basically represents that these are readily effluxed by the P-glycoprotein. So they have an affinity for the P-glycoprotein, and even if they're taken up in the CNS, it is effluxed out. But anyway, so with this we could find that these all can be used as drugs, which can be readily bioavailable. So this is, so what we did was that then you have the Z and analyze code. In what you do is that you take the different descriptors, property descriptors, and convert them into binomials of 0 and 1, and then try to generate a normalized ZI score. And if we look at the ZI score, we look at this compound 1.81, and it should be more than 0.56 to be a good drug. So we found that the compound 3, 1, and 4. So this is the 3, this is the 1, and this is the 4, these have a higher value comparatively, and this is in comparison to the other widely available chemotherapeutic drugs which are available in the clinic. So next thing which we wanted to do is that we wanted to check whether this binds with the DNA or not. So what we did was that we took ctDNA, and we did the absorption spectral studies of organotin compounds with ctDNA. This is the different concentrations of ctDNA which we have used. So what it shows is that these compounds, so when the compounds react with this, with bind with the DNA, there is a hypochromic shift. And then there is a hypochromic shift that tells you that it is interacting with the DNA. So that is what we found in all our compounds 1, 3, and 4. We also did the Bernoulli-Hilbrand intercept to slope ratios and to calculate the K values. And these K values are also within the normal range, again suggesting that we are onto different good compounds. And these are the delta Gs, which again shows that the binding is efficient and binding is suitable. So then we looked at the molecular docking analysis of organotin compounds, And now this is the DNA, and these are the different compounds, 1, 3, and 4. These are the ones which we went further with from the Swiss Agnes reports. Now these ones, they interacted with DNA through hydrogen bonding. This green is the hydrogen bonding, and then hydrophobic interactions are the red ones. So all of them they interacted with the DNA. Then, to confirm this, we moved on to the fluorescence emission spectral studies of these compounds with ctDNA. And if you look at the data, compound 1, 3, and 4, if you look at compound 3 and compound 4, it leads to a decrease in the intensity of the emission. And once you have the decrease or hypochromic shift, which is called, that means it is intercalating with the DNA. It is an intercalating molecule with the DNA. While compound 1, it was not so clear, and compound 1 was the one, so we thought that compound 1, rather than having an intercalator between the different stacks of the nucleic bases, it may be acting, or it might be acting through the groove binding mode. So, to prove this further, we took 2 dyes. One was ethidium bromide and second was the hex. So ethidium bromide is an intercalating DNA, and while hex is a groove binding. So we did a comparative studies between them. And if you have the ethidium bromide DNA system, then ethidium bromide, if you look at again compound 3 and 4, so ethidium bromide DNA contacts the fluorescence, and when my compound is going to bind with that, there will be a decrease in the fluorescence of ethidium bromide DNA complex. So they had the hypochromic shift, and which proves that these are 2 intercalating only. So these are intercalating molecules. While if you look at the hex, in the hex, compound 1 did not do so in this. While when we treated with the hex, then in the hex, it's a groove binder. So here the compound 1 was interfering with the binding. So that proves that it is the groove binding. So compound 3 and 4 were the intercalating molecules, while compound 1 was a groove binder. Then we did the molecular studies of organotin compounds with BSA, because anything or any drug which has to be carried in this body has to bind with the protein to be transferred. So we looked at the BSA molecule, and then we found that these are the interactions, these are compounds. All of them they combine with 2A and 3A domains of subdomains of BSA, and they also have showed the hydrophobic interactions and hydrogen bonding with the BSA. Further, we have then looked at the fluorescence emission spectral studies of organotin compounds with BSA, and again with compound 3 and 4, and all of them actually, in fact, they had a hypochromic shift and they lead to a decrease in the absorption. We also looked at the scattered, this is Stern normal plot, which is here, Stern normal plot, and then we also did the scattered. So Stern normal, if you look at this, these are the different compounds, and if you look at the compound 1, compound 1 is this, and compound 4 is this one. So there is going to be a static quenching, there is going to be a dynamic quenching. What was found that compound 1 was having only static quenching, while because of this increase in the binding constants which we found out, it exhibits it as both dynamic as well as static quenching with the BSA molecule. We also calculated the number of binding sites and scattered plot constants and so on with this. So further we wanted to now prove it that yes, okay, these are the compounds, and we look at whether they are anti-cancer molecules or not. We have to treat it with the in vitro. So we took different cell lines. We took 3 cell lines. We took HCT116, we took highly metastatic SW620, and then we took a normal epithelial cell line also, because to see that whether the effect is cancer tumor specific or does it have a similar kind of effect on the tumor, on this normal epithelial cell also. So IC50, if you look at it, this is we have the IC50 for 31 for compound 1, compound 3. Compound 3 was highly cytotoxic. While compound 1 and 4, they are of this 31% and 21% in this case, 5.663 and 6.75% in this case, and with this also there was a cytotoxicity which was observed, and these were the corresponding values for the cisplatin and 5-fluorouracil. Then we looked at the apoptosis, measurement of apoptosis by these compounds, and we did the annexin 5 PI complex with different cell lines, and so this is what happens. So there is so this is the one that, so if you look at that, there is an increase in the early apoptotic cell lines, and cells and the late apoptotic cells with compound 1, compound 3, and compound 4. All the 3 compounds, they could lead to apoptosis. So this is with a different cell line, a similar story here also. And then we moved back to cell proliferation and we looked at the compounds. So this is the cell lines. This is the percentage of cells in the gene of G1, this is percentage of cells in S phase, and this is the percentage of cell in G2M. And again, there was a G2M arrest in all these groups. If you look at this, this is the cisplatin which we did here. Cisplatin was also leading to G2M arrest. So this is again the SW620 cell lines, and so again the same kind of a data we got. So finally, I would like to conclude my talk that here we synthesized 5 compounds, which from 1 to 5, and all in this one-pot reaction at room temperature. Overall yield of these compounds was almost 80% in all cases, and except for compound 5, all other compounds, they showed a good ADME profile or ADME profile, and based on this we decided to move further with 1, 3, and 4, and then we looked at the spectral studies to show that compound 1 is a groove binder and compound 3 and 5 are intercalators with DNA. While we did with BSA, we found that it is, this is the fluorescence spectrum, and molecular docking studies also confirmed that they also bind to the BSA, all the compounds, and all of them, they displayed a good cytotoxicity against colon cancer cell lines, and all of them, they were able to induce the G2M arrest and also promoted apoptosis. So how are we going to go further? Because the story started whether it is targeted or not. So this is just a part of this. We are continuing with the work, and how we are moving ahead with this is that we are going to do the in vivo studies with different doses of these 10 compounds which we have selected, and then we look at the tissue biodistribution, how much tin is going at what ways to check that whether there is any toxicity or not, and also the overall toxicity profile in the animals. So this is where we are going, and this paper should also be coming out very soon. So this is my collaborator, Dr. Varun, who is from Department of Chemistry, Punjab University. This is the student, Dr. Vinay, who was a D.S. Kothari Fellow in my lab and now has moved to Harvard Medical School. And this is my lab, and this work is done by Shagun. For this project, we got our funding from CSIR, but we also have grants from ICMR, and so, and we would like to acknowledge our grant funding agencies for that. Thank you so much, and open to the questions. Yes.
Do you think if you were to do combination, 1 and 3 or 1 and 4,
yeah,
because you have 2 different kinds of cells,
yes,
that might give you a better effect?
You are right, it may, but this toxicity also increases because what happens is
when you do combinations,
dose goes down, yes. What we observe was that when we were standardizing, that I am showing the in vivo data, that when we use a higher concentration of any tin derivative, it leads to lethality. So we are using very, very small doses in this, and we haven't tried the combination one because right now we're just looking at this 1, 3, and 5, which we decided to 1, 3, and 4, which we decided to proceed. But yes, that's a good point. I can have a look at that also. Yeah.
Any other question for Dr. Agnihotri? No, thank you very much, ma'am.
Thank you.
Nice talk.
Thank you.
I would like to ask Professor to please state Professor Navneet for the such an insightful talk. Professor Bhushan.
So may I have the last speaker, Dr. Ajay Kumar. Dr. Kumar is from Department of Zoology, Banaras Hindu University, Varanasi. He has been working in the area of cancer biology. He is also PhD from BHU, and he received Dr. D.S. Kothari postdoc fellowship and worked at Emory University, Atlanta. Now he is assistant professor at Center of Life Sciences, Central University of Punjab, and he is also member of European Association of Cancer Research, Indian Society of Cancer Biology, and European Federation of Biotechnology. Dr. Ajay Kumar.
Yes, yes. Thank you, sir. Thank you for the nice introduction. So, first of all, I would like to thank organizing committee, especially Professor Alok Chand Bharti and Professor Anju Srivastava for giving this opportunity to present my work here on this platform. So, the title of today's presentation is Therapeutic Potential of Nimbolid against T-cell lymphoma, a role of altered glucose metabolism, pH homeostasis, and oxidative stress. So, first of all, I would like to briefly discuss about the lymphoma current therapeutic strategy and classification of that. So, lymphoma, it's a cancer of lymphocytes, particularly T cell and B cell, and that basically originates in lymphoid tissue. We can say that the organs like bone marrow, thymus, spleen, and lymph node. So, it is the 7th most common cancer. And ranked 9th for cancer-associated death worldwide, according to Globocon 2018, lymphoma is the 11th most common cancer in India, and it is estimated that the burden of lymphoma is going to be doubled by 2035. So basically, it is classified into two groups. One is broadly lymphoma into two types, T-cell lymphoma and B-cell lymphoma. T-cell lymphoma is rare and constitutes 10 to 20% of total lymphoma, and the prognosis is very poor, and that's why the survival is very less in comparison to this B-cell lymphoma. So therapeutic approaches are basically immunotherapy, bone marrow transplantation, radiotherapy, and chemotherapy. Like other cancers, still chemotherapy is the first line therapy for the treatment of T-cell lymphoma. And but the major drawback of using whatever the anti-cancer drugs those are available, they causes so many of side effects like alopecia, cardiotoxicity, nephrotoxicity, and vomiting, and peripheral neuropathy, reduced fertility, and all. And that's why in recent past years, phytochemicals have emerged as a better choice of anti-cancer drug due to minimum or no side effects, easy availability, low cost, and multiple targets. So, and more so, in recent few years, approximately 50 to 60% of cancer patients in USA are being treated with compounds, either they are derived from the plants or from the nutrients. So, plants with the medicinal importance, this neem, also called Azadirachta indica, is the most common traditional medicinal plant, which different plants which different parts have been used for the treatment of various diseases well known established from thousands of years. Okay, so hence it is popularly called as village pharmacy in Asian countries, including India. And interestingly, more than 30 bioactive molecules are isolated from the different parts of the neem, like leaf, roots, flower, and all. Including azadirachtin, epoxy, azadirachtin, nimbin, quercetin, nimbolide, which have shown promising tumoricidal action against various cancers. So, one of the studies conducted by Cohen et al., they found that among several limolides, nimbolide exhibits maximum cytotoxic potential. More so, a study conducted by Cheetah et al., they found that, and they basically identified that the BCL2 is the major target. So, due to all these things, we selected this nimbolide for our study, and accumulating evidence suggests that nimbolide exhibits its tumoricidal action by inducing apoptosis and inhibiting various crucial cellular processes like invasion and metastasis, angiogenesis, and cell proliferation. But still, nothing is known about how this nimbolide going to alter reprogrammed glucose metabolism and tumor acidosis. These two are the major hallmarks features of the cancer cells. More so, very little is known about the tumoricidal action of this nimbolide against T-cell lymphoma, really complex neoplastic disorder. In terms of the clinical management. So that's why we started working on this T-cell lymphoma. And as we can see from here, these normal cells, they show more dependency on oxidative phosphorylation for their energy requirement, whereas in case of transformed cells or cancerous cells, neoplastic cells, they basically going to get going to obtain the energy from the glycolysis. Although from the glycolysis they are only going to get 2 ATP. The reason is that cancer cells are, as we all know, that these are the highly proliferating cells, and they need quick energy. More so, this is the pathway glycolysis, which is going to provide various precursor molecules of macromolecular synthesis. Like here, we can see that this glycerol 3-phosphate is involved in the nucleotide synthesis. This 3-phosphoglycerate in serine biosynthesis. So this is the region the cancer cells switch their metabolism from oxidative phosphorylation to this glycolysis. So, and usually they do this by elevating the level of various glycolysis regulatory molecules like glucose transporter GLUT3 and then PKM2, LDHA, lactate dehydrogenase A, PDK1, pyruvate dehydrogenase kinase 1, and monocarboxylate transporter 1. So, apart from this, cancer cells produce large amount of acidic metabolites, including this lactate, which is an end product of aerobic glycolysis. Because cancer cells, the reason is that cancer cells perform glycolysis even in presence of oxygen. Usually normal cells that do this only in the absence of oxygen, and that is nothing but the Pasteur's effect. But in the case of cancer, they perform glycolysis even in presence of profound oxygen, and that's why that is called Warburg's effect or aerobic glycolysis. It's play a crucial role in developing or creating an acidic tumor microenvironment, and that is the reason this. In case of cancer cells, reverse pH gradient has been noted. So in case of cancer cells, what we can see that the intercellular pH is more than 7.4, whereas in case of normal cells, it is near about 7.2, whereas extracellular pH is very low. It's near about 6.7 to 7.1. So why they do this? The benefit of this reverse pH gradient is that this higher intracellular pH is going to inhibit apoptosis, so they are going to protect them to undergo the apoptosis. More so, this acidic tumor microenvironment, they basically, this acidic tumor microenvironment promotes immunosuppression. They are going to suppress the function of various immune cells like macrophages and all, and also promotes invasion and metastasis by activating MMP, matrix metalloproteases, and induces apoptosis in case of normal cells. So that is the reason. So many benefits. That's why cancer cells basically create this acidic tumor microenvironment. And in creating this acidic tumor microenvironment, various pH regulators play a crucial role. Some of them are NHE, sodium proton exchanger, BATPH, and then MCT monocarbonylate transporter, which is basically a transporter which is involved in the transportation of lactate, which is going to be produced due to the aerobic glycolysis by the cancer cells, and then carbonic anhydrase and all. So, due to mentioned about lactate, we designed two objectives. In the first part, we are going to basically evaluate the anti-tumor activity of Nimbolide against T-cell lymphoma in vitro and in vivo. And then in the second part, I will show you some of the interesting results regarding the effect of Nimbolide on the programmed glucose metabolism and tumor acidosis. So, in the first part, what we did, we firstly performed MTTSC on some of the T-cell lymphoma cell lines. One is DL cells, Dalton's lymphoma cells, and HAT78 and J6 cells. Dalton's lymphoma, it is a type of murine T-cell lymphoma, whereas these two, HAT78 and J6, these two are the human-originated T-cell lymphoma. And as we can see from here, that this Nimbolide inhibited the survival of T lymphoma cells in both the cases, whether these are of murine origin or human origin, in time as well as in dose-dependent manner. Further, as we know that apoptosis is one of the major mode of cell death, we estimated the apoptosis with the help of Annexin-5 and TAP staining, and as we can see from here, Nimbolide induced apoptosis and more apoptotic cell population were noted in case of Nimbolide treated T lymphoma cells. Further, we confirm this at molecular level, and as we can see from here, the expression of BCL2 gets down-regulated where the pro-apoptotic molecules at PF3 bags and cleaved caspase 3 were upregulated. The same we found in the case of HAT78. Here we did not check the PF3 level because it is mutated in the case of HAT78. So further, we moved to the in vivo model and did some experiments. We used 0.5 mg per kg body weight Nimbolide, and then as we can see from here, so the ascitic fluid is going to be decreased. So peritoneum load is going to be decreased here as compared to control. More so, as we can see from all these three bar diagrams, that the body weight decreased in the case of Nimbolide administered tumor-bearing mice, ascitic fluid that is also decreased, and tumor cell density we also found decline. So apart from that, what we noted that the tumor cells, those were isolated from Nimbolide administered tumor-bearing mice, in that case, survival was inhibited, significantly, remarkably inhibited as compared to the control group. So all of these results suggest that obviously Nimbolide not only inhibit the tumor growth in vitro, but also in vivo model. Okay. So similarly, we also checked apoptosis by NXN5 and DAPI staining, and we got the same kind of result, induction of apoptosis. In case of tumor cells, those were isolated from the nimbolide administered tumor being mice, and similar kind of pattern we found in the case of apoptosis regulatory molecules. Further, ROS reactive oxidation species is found as one of the prominent factor in nimbolide mediated apoptosis, and that's why we check the level of ROS. As we can see from here, the ROS level was highly elevated in case of nimbolide tumor cells that were isolated from the nimbolide administered tumor mice. And as we all know that superoxide dismutase and cartilage, both of these two play a crucial role in regulating the pH homeostasis. We checked their level and found decline. So obviously, the apoptosis which is going to be induced by nimbolide, that is probably through the ROS mediated. So apart from that, we also checked the effect of nimbolide on crucial cellular and non-cellular components of the blood, particularly total leukocytes, lymphocytes, monocytes, hemoglobin, and RBC. And as we can see from here, this is the normal in the case of the normal, and this is the control group, and this is the experimental group. So as we all can see that nimbolide treatment tried to recover the leukocyte number and lymphocytes and monocytes, which was found altered in the case of tumor-bearing mice. So all these results suggest that it can. Control means are totally normal, means nothing, no tumor. No, this normal is the nothing, means no tumor. Control is having only tumor being no and administered with the Vital only. Yeah, without treatment. Exactly. So in the second part, we check the effect of nimbolide on two crucial hallmark features of cancer cells, that is the reprogrammed glucose metabolism and tumor acidosis. So this glycolysis, as already I mentioned that the tumor cells, most of the tumor cells show dependency on aerobic glycolysis, and this is one of the crucial metabolic adaptive feature. And we check this by examining the level of glucose, lactate, and pH. And interestingly, we found increased level of glucose and pH, whereas decline level of lactate in the extracellular region, which clearly suggests the anti-glycolytic activity of nimbolide. Apart from that, we also check the level of crucial glycolysis regulatory molecules, glucose transporter group 3, PDK pyruvate dehydrogenase kinase 1, which is known as a gatekeeper enzyme. So this is basically involved in phosphorylation of the pyruvate dehydrogenase, and by doing this, it's blocked the entry of the acetyl-CoA into the TCA cycle, and thus forces the tumor cells to do glycolysis even in presence of sufficient oxygen. And here we can see that nimbolide inhibited both this GLUT3 and PDK1, and this HIPAA alpha is the transcription factor for both of these molecules, and that is also found down-regulated. So probably nimbolide is inhibiting or suppressing the expression of GLUT3 and PDK1 through the HIPAA alpha. The similar kind of pattern we also found observed in the case of 78 cells. So then we moved to the in vivo experiments, and we found again the elevated level of glucose, declined level of lactate in the acidic fluid, and then the decreased level of GLUT3, LDHA, lactate dehydrogenase A. So as I mentioned that this is in the generation of or creation of acidic tumor microenvironment, pH regulatory molecules play a critical role. And as we found that the this extracellular pH means pH of the acidic fluid was found elevated in case of nimbolide administered tumor being mice. We checked the level of pH regulatory molecules like monocarbonylate transporter 1, NHE1, carbonic anhydrase 9, and BATPH, and these all were found down regulated. So this could be the reason. That's why the level of extracellular pH was found elevated. So overall, the conclusion is that nimbolide triggers apoptotic cell death in T lymphoma cells by altering the levels of apoptosis regulatory molecules. Nimbolide exhibits anti-glycolytic activity by modulating the expression of glycolysis regulatory molecules, including transcription factor heparin alpha. Nimbolide has also the potential to reverse the acidic tumor microenvironment by suppressing the expression of pH regulatory molecules like MCT1, NHE1, carbonic anhydrase 9, and BATPH and all. Hence, nimbolide-related glycolysis inhibition and reversal of acidic tumor microenvironment might have also a crucial role in tumor growth inhibition. Overall, the experimental findings of the present study demonstrate that nimbolide might be a promising chemotherapeutic drug for the treatment of T-cell lymphoma and may play a significant role in designing of the novel chemotherapeutic regimens against T-cell lymphoma. So, this is my small research team, and the work which I presented here was done by Pradeep Kumar Jaswara. My lab is supported by University Grant Commission and then SCRB, ICMR, and this the work which I have shown here basically was supported by UGC. Thank you all for being here and listening to this.
So, any question for Dr. Kumar? Yes.
Thank you, sir.
So I want to know, you try to have that this topic answer was right, normal activity.
Sir, actually, we treated the mice, tumor being mice, for 12 days, and we found that most of the mice were surviving more than 50 days. So I think even because we only administered the nimbolide only for 12 days, and even though survival was very fantastic, so I think it will be having a great potential. What? Yeah. But the problem was we did this, but what we found, because the killing potential of this nimbolide is much better than even the cisplatin, and that was the reason when we are administering both cisplatin and nimbolide, there was no much difference. That's why we drop out that the idea might be in future we will plan something, means why it's happening. So probably because nimbolide itself at very low dose was, and in case of, it has been observed that in the case of leukemia, nimbolide so basically the cytotoxic ability at very low dose. So
My last question.
It's okay, sir.
So like all this, there is a lot of kind of
So, but yes, yes. Yeah.
What is nimbolide? Do you have anything directed?
Sir, it's really a good question. As I have already shown you, I have here shown that there was nothing, means even the number of lymphocytes, monocytes, that's all increased. So there is no any side effects. Apart from that, I have not shown some of the data, and then where we did some histological analysis and found that this nimbolide treatment is going to reverse the cytotoxic effect of this, means the tumor burden due to the tumor, whatever the changes are going to be happening in the kidney and on the liver, so that is going to be reversed by the Nimboda. So
I think you should have used normal cells.
Yes, sir, we did.
To show that it has no problem.
Yeah, we did.
That is number one.
Yes, sir.
Number two, you should have a positive control with a non-anti-cancer.
Yes, sir, you are right.
Not in combination, not
Yeah. You are right.
correctly.
Yeah, you are right.
So that would have.
Yes, sir. But none of them ever asked
I would have.
Yes. It's fabulous. Okay, thank you for the service.
Any more questions? So thank you very much.
Okay,
Dr.
thank you,
Kumar.
sir.
And on behalf of organizers of INCT 2022 and on my own behalf, I thank all the speakers for their best effort and all the audience to have so much patience. Thank you very much. Have a nice day.
I now request Professor Kumar for giving such an insightful lecture.
Wants a picture with this, so I think we can just one more second.
And finally, I would like to request Professor Dinesh for being the chair of this session.